So, place yourself in the 1980s. This is kind of hard because, as a scientist, you had just found out that retroviruses could infect humans. Now you
Hmm.
find out that HIV is a retrovirus. You're like, wait, I didn't even know that RNA could be turned into DNA until 10 years ago. So that's quite a tricky position to be in. Making effective new medicines isn't easy. Welcome to Hard Drugs. I'm Saloni and Dattani, a researcher on global health at Our World in Data, and one of the founders of Works in Progress magazine. And I'm hosting this podcast with Jacob Trefethen, who leads science and global health R&D funding at Open Philanthropy, and is one of the most fun, interesting people I know. This show is about medical innovation, how to speed it up, how to scale it up, and how to make sure life-saving tools reach the people who need them most. It all started with a conversation, a shared instinct that this was the right time to start a podcast to dive deep into how technologies for malaria, cancer, AIDS, and other diseases actually came to be.
Today, we're going to talk about HIV. Making an HIV vaccine has been the holy grail for many of the world's top scientists over the last generation. It has proven one of the most challenging scientific problems too, and we don't yet have a vaccine. But last year, one drug company announced they'd gone a completely different route. They made a drug you get injected with once every 6 months or maybe only once a year, like a flu shot, giving you almost perfect protection against HIV. So, how did we get here? And what does it mean for one of the world's deadliest diseases?
Okay, I'm super excited to talk about HIV, Lenacapavir, and other HIV drugs today with Jacob. Hello.
Hey, how you doing?
Great. Yeah, so I'm super excited about this. I think we have a bunch of things that we're going to talk about in the episode. Maybe starting with just what HIV is, how it infects people, and then moving on to the history of drug development in the field, how lenacapavir was developed, what lenacapavir actually does, and then where we are now, how to scale it up to people who need it.
That sounds good to me. And I feel like I first really heard about or realized what a big deal Lenacapavir was from a tweet from you. So I get to be the lucky guy who gets to be taught some of that history of HIV and how it all fits together from you. And hopefully I can chip in some of my knowledge from working at Open Philanthropy on global health R&D as well. So that's this conversation. There's a lot to cover. I hope we cover the stuff that matters and get to the finish line of what are these magical new drugs really doing.
So there are a lot of, I think, subplots that we're going to go through. The whole process of drug discovery in this field has been really amazing. If you think about what HIV was like in the 1980s, where people would only have a few years of survival after being diagnosed, to now, where if people take treatment early enough, they can expect to live almost a normal life expectancy. And I think talking about how that's happened, how that's been made possible, is really important.
I agree. And it sounds like we're going to have to start with what the heck is HIV itself? Shall we begin there?
HIV is a virus that causes AIDS, the acquired immunodeficiency syndrome, which is associated with lots of different infectious diseases, cancers, and conditions that people suffer from if they've been infected with the virus for long enough. But what's interesting about HIV to begin with is that it's not a typical type of virus. It's something called a retrovirus. Retrovirus itself is a new concept to a lot of scientists, historically speaking. The first retrovirus that infected humans was only discovered in 1979, which is just 2 years before the first reported cases of HIV in the US. Before that,
Yeah, that's
people
so.
had no
And that was
idea.
not HIV. Okay, that was a
That was
different
not
retrovirus.
HIV.
Okay.
That was human T lymphotropic virus, the first human retrovirus discovered. And I think that itself has a really interesting story. So when I was first reading about this, I was really struck by the fact that there was only this 2-year gap. We essentially figured out what retroviruses were in the first place just shortly before discovering this deadly new disease that was caused by one. And I think that's an important thing to think about when we're thinking about what scientists at the time would have been working on and how they would have figured out that it caused AIDS.
What is similar about that virus and HIV? What makes it a retrovirus?
So, a retrovirus, maybe I could kind of step back a bit and talk about how the usual process of DNA works, just to give you some context.
Great.
So, almost every cell in our body has DNA. DNA is the genetic code to tell us which proteins to make, but it contains all of that genetic code. And all of our cells don't need to be producing all of those proteins, and they don't need to be producing them all of the time. So instead of using the entire genetic code, we use our enzymes to find segments of the DNA to turn into RNA, which is this intermediate molecule that's also used for various other things. And then we turn this RNA into proteins that are used in all kinds of processes in our body. So this direction from DNA to RNA to protein was how biologists and scientists typically understood cells and biological life worked. And that was
Right.
overturned with the discovery of retroviruses. So what happened here was that people found out that there were certain viruses that could turn their RNA into DNA using an enzyme called reverse transcriptase. This
Go in the
was
other direction.
yeah, so this was a huge discovery in 1970 by Howard Temin and David Baltimore. They each independently discovered this enzyme, reverse transcriptase, in chicken and mice. They discovered that retroviruses could reverse transcribe RNA into DNA. And then they tried to find other retroviruses that infected humans. For a long time, no one succeeded in finding any of these viruses until 1979. So it was about almost 10 years of people trying to find real examples of these, and they couldn't.
And is that related to HIV in 1981? You know, we just got lucky, or yeah, why, why so close?
So I think we did get lucky, and I think it would have been really difficult to figure out that
Huh.
HIV was causing AIDS if not for that, or to even know where to look.
Wow.
And so what's really interesting about this is the scientist who discovered the first retrovirus that infected humans, Robert Gallo, was also one
Right,
of
I
the
remember.
scientists who discovered HIV as being the cause of AIDS. And I really like kind of reading through reviews or like retrospectives written by scientists themselves on how they figured out something, what research they did. And so I was reading this retrospective that he wrote on the discovery of retroviruses and HIV. And it was really interesting because he talks about how people were really skeptical that there were any other retroviruses that infected humans.
Mm.
And the reason for that was they had people had found retroviruses that infected other animals, other primates, and in those primates it seemed to be pretty abundant or like ubiquitous across their organs, but that wasn't the case in humans, and it was just hard to find them. So they assumed that maybe there's something that prevents us from being infected by them. And scientists also found that if you put the animal retroviruses into human blood, human serum, that would immediately inactivate those viruses using our complement
Okay.
system, which is part of like one component of our immune system. And so this suggested that maybe there's some way that we're just protected against them. They're not going to affect us. And I think Robert Gallo just had this idea that that might be wrong. Maybe there are some other types of retroviruses that we hadn't studied. And so he started looking at T cell cancers. So in animals, the retroviruses that infected those animals would typically cause T cell leukemias. So T cells are a type of white blood cell. And so he thought maybe they're also causing leukemias in humans. And he started working on finding patients with T cell leukemias. Eventually, he did actually discover a retrovirus in them. So this retrovirus was using reverse transcriptase to turn its RNA back into DNA. And the reason that this is important for HIV is that they actually developed the tools to test out whether there are retroviruses infecting a sample. As part of that process, they started working on potential drugs that could be used to target reverse transcriptase. They also just generally
Hmm.
had the idea that humans can be infected by retroviruses, and
Right.
retroviruses infect humans in their T cells. And as we'll come to later on, that's very relevant in HIV.
Okay, so just stepping back for a second, the beforehand scientists thought, well, maybe these retroviruses that we're seeing in other animals aren't infecting humans, and the human immune system's basically winning against them. We've got this under control. But then it looked like they may infect some cell types, and we might not be winning fully. I mean, my question then is, what it sounds like such a good strategy for a virus. If I'm a virus, I would love to reverse transcribe and integrate in your DNA. So how come if it's possible for HIV, we don't see this in lots of viruses? I mean, are there other viruses other than
Yeah,
HTLV you mentioned? I mean, how come we're not there? Not lots and lots.
that's a really interesting question. And retroviruses, it turns out, are really ancient. Parts of
Mm,
their genomes are integrated into our normal DNA, and they've just been passed down over time. So this is called endogenous retroviruses. I don't really know much more than that about that
wow.
topic. But on your question on why aren't there more retroviruses infecting humans, I think there's potentially three or four things going on. I'm sort of wary of saying that something's not possible because sometimes people say that even in the case of this, and then they figure out that it's wrong.
Right.
Maybe we just haven't found those
Yep.
other retroviruses. But so some of the reasons probably one, it's like an error-prone process. If you're so
Hmm.
you're a virus. Sorry, you're not a virus, but for a
Thank you.
virus, for a virus that's transcribing its RNA genome into DNA, the reverse transcriptase enzyme is not very precise in how it does that. It introduces errors into the
I
code.
see.
That's
Right.
probably a little bit dangerous for the viruses themselves.
Got it.
Secondly, they have this RNA genome. They then transcribe it into DNA, and then they move the and then they get our cells to transcribe the DNA back into RNA, which just seems a little bit inefficient, right? It would just take a longer amount of time. It introduces errors.
I think.
That's not super useful, maybe, but obviously in some cases it actually is, and it's worth that trade-off.
You mentioned T cells. It's going after these. I mean, this is really it's clever, but it's creepy. So there's a reverse transcription where it's then gonna integrate into my DNA, which is disturbing in its own way. And then additionally, you're telling me it's going to do that not just anywhere, but in one of my immune cells, which is what's meant to be fighting infections. It's going to hijack and integrate there. So is that right? And those are the T cells.
That's right. So HIV infect um, various immune cells, but usually a specific type of T cell called a CD4 T cell. And the CD4
CD4,
is
right?
the CD4 just describes one receptor on the surface of this T cell that is very crucial in signaling, but also kind of defines that type of T cell. And these types of T cells, I guess I should just describe what those actually are. So we have different types of white blood cells in our body. T cells are an important type. In this case, what they do is they help the body recognize pathogens or things that we've seen before by presenting parts of that pathogen to our other immune cells that can last much longer in the body and remember them if they appear ever again. So HIV is essentially infecting these quite important white blood cells in our body.
So what happens then? We've entered the cell. Why is that a problem? How does it cause disease?
We've entered the cell. Well, so there's quite a long process. So initially, when people get infected, they have this short-term infection, some kind of fevers, flu-like symptoms, things like that, and the virus quickly replicates itself, multiplies into many copies. Those copies then infect other T cells. They then go into our lymph nodes, which are basically these little hubs of immune activity. So there's some in your neck, some under your arms, other parts of your body. So it infects these different immune related tissues and depletes them. And that means that people become more vulnerable to all sorts of other infections that are normally mild to people, or just, infections in general, and some cancers. So our white blood cells are also useful in detecting tumor cells and trying to eradicate them. And by depleting those important cells, people also have a higher risk of certain cancers. So we have this short-term infection that immediately depletes a lot of our immune cells. Eventually, there's this kind of slowdown in how much it replicates, and you get to this equilibrium. But that equilibrium is still much worse than if someone hadn't been infected. And so over a long period of time, this this reduction in immune cells means that people are vulnerable to various diseases. And as time goes by, they get sicker and sicker from those other infections.
And before HIV, were there infections that were known to cause cancers? Because that's now something that we're more familiar with. But was that connection a surprise and made it harder to figure out what the real cause was?
HTLV, the first retrovirus that was discovered just 2 years before, that was the first pathogen that was clearly causing cancer in humans. After that, there have been a bunch of other pathogens that are now known to be cancer causing. So, one is hepatitis B, which you do a lot of
Yep.
research on. HPV, human papillomavirus, that causes various cancers, including cervical cancer. There's Helicobacter pylori, this bacterium that causes stomach cancer. I think there's more. Maybe you remember some others. And there's HTLV, which
Hepatitis C.
causes...
Yeah.
Oh, hepatitis C. That's right.
Yeah. Those hepatitis viruses sneaky.
Yeah. So coming back to your question, the cancers were the first, I think, surprising thing about people who had AIDS. So one of the types of cancers that they became vulnerable to was called Kaposi sarcoma, which is this tumor. What was surprising about that was usually doctors who saw patients with this type of cancer would see them in quite elderly patients or people with, I don't know,
Yeah.
severe immune deficiencies and things like that. And in this case, they were seeing them in young adults, which was really surprising. They were also quite severe cancers. They were hard to treat with the usual treatments that were available. Um, and the fact that this was growing in prevalence was also really surprising and worrying to people. So I think that was one of the first noticeable kind of warning signs that there was some kind of epidemic spreading. It was probably an epidemic disease that was caused by some pathogen. I think there were maybe like a few months or a year or so before people realized that it was probably caused by a virus. And I think the reason for that was that there were some cases of people being infected through blood transfusion. So they had no other connection to other people with the disease. And they
Mm.
had no other environmental risk factors or anything like that. But they had recently had a transfusion or an organ transplant or something like that in the meat, and then suddenly they got infected. And the reason that this links to being a virus is because you can usually filter out or purify some of the blood that you're using or the organs with filters that get rid of bacteria, which are bigger. But that doesn't always work for viruses, which are much smaller. And so the viruses were getting through, and they were still infecting people. And the
Uh,
fact that
right.
this was infecting people far away with no other connection suggested that it was a virus.
I see. I see. I mean, maybe that's a good point to just talk a bit about transmission. So how does a virus transmit? I've got some ideas, but
There are different types of modes of transport. So one is, as we just talked about, blood transfusions and organ transplants. So if there is contaminated blood with HIV, the risk of an infection to someone else is quite high. It's some. I was looking at this data from the systematic review, and they estimate that the risk of infection is about 92% from a transfusion of contaminated blood, which is quite high. Mother to child transmission. So mothers who are infected with
yeah.
HIV had a quarter of them would pass HIV down to their baby.
Oh,
And this was
yeah.
before treatment was available. So now
Mm,
the chances are much lower if people use antiviral treatments around the time of pregnancy and childbirth. But that was obviously very
yes. Yeah.
scary. And
Yeah.
then sexual transmission uh, is probably the most common route that most people have heard of for HIV. And then finally, injection and drug use. So using shared contaminated needles with HIV.
So before drug development, how come we need drugs? How come the immune system doesn't control HIV better? I mean, I have a stereotype that it's extremely hard to control. It's quite if an infection gets established, it's really tough for us. So why is that?
One really interesting thing that I learned while trying to read about this was that HIV is usually caused by a single virus particle. Um, and that one particle replicates enough that it can cause a whole
Oh, you're
infection. And
scaring
so
me, Saloni. That's scary. That's scary. So do you mean that one particle is one particle entering my bloodstream is all it takes, or you mean something else?
I actually mean something else. So
Okay.
if you look retrospectively at people who have HIV, you take a sample of their blood and you look at the different virus particles. If you then trace back the genetics of those virus particles, you then find that they all have one common ancestor in around three
I
quarters
see.
of cases.
I see. I see.
But that
Okay.
doesn't necessarily mean that just one particle is enough to infect someone because we have enough barriers in our immune system. I mean, if you think about like a skin infection or something, we have our actual skin, we have several layers of skin, we have immune cells protecting us within our body. There are various barriers that prevent bacterium or something from infecting us, and that's the same is
Hmm.
true with HIV. So I think what's happening here is that if you think about this from the perspective of probability, there are many barriers, but eventually one of them might be able to cross all of those barriers and
Got it.
cause an infection. And if it's able to do that, it can replicate very quickly. And I think that gets us to why it's difficult to control an infection. So even though we do have these barriers, HIV is just very fast at replicating, mutating. The reason that HIV can mutate so quickly, basically becomes really genetically diverse within a person who is infected. And that means that our immune cells might be able to recognize some of the HIV strains that are in our body, but it's very difficult for it to keep up with the rapid evolution and the
Mm,
increased diversity. But there are several reasons for that. One is the reverse transcriptase enzyme that we talked about that turns RNA into DNA. That enzyme is not very precise, and so that introduces errors. The errors allow it to potentially get beneficial mutations sometimes, and that means that it can kind of genetically diverge. The
mm.
other is that the HIV particle has two copies of RNA inside it, and those two copies can recombine with each other.
Wait, hold on. What? It comes in with two of the same thing?
It, two of almost the same thing. So it has two.
Okay.
So in the same way that we have two sets of chromosomes in our cells, it
Okay, fair enough. I
has
guess that's true,
two.
yes. Oh,
It has
wow. Okay.
two
That
copies of
that
RNA inside it.
they're not it's not a double-stranded RNA. They're
No,
separate
that's right.
strands.
They're two
Wow.
single-stranded RNA virus, two
Huh.
single-stranded RNA particles, and they can get transcribed. They can get reverse transcribed separately. They can also recombine with each other. And so I was reading this
Wow.
review paper about how all this worked, why there was such rapid mutation, and they said they were talking about this recombination, and they said this can be considered a primitive form of sexual reproduction. And I was
No, no,
mind
no,
blown.
no. This is a virus. If there's one thing I know, it's that that is too complicated. I don't believe this review paper.
No, it's
Okay.
crazy. And then there's a third thing, actually, which is that our enzymes introduce errors and mutations into HIV. So we have this family of enzymes called the ApoBec family. They insert mutations into HIV to try to damage it. So what they do is they change the G's in our bases in our DNA
Hmm.
into A, and they do
Okay.
that on a single-stranded DNA particle. So the HIV virus has these two single-stranded RNA molecules. They get turned into DNA. Then our enzymes introduce errors into it by turning some of the G's into A's.
So just taking a step back, you're saying that the virus itself mutates a lot. So the reverse transcription stage introduces errors, and that actually, in a sense, helps the virus evade our immune system. So it is introducing errors that make it hard for us. And then you're saying we also are introducing errors that make it easier for us, or rather harder for the virus. So we're both sort of fighting fire with fire of I'm going to make you different, and it's saying no, I'm going to make myself different, and the we're in a kind of ratchet situation there.
Yeah, it's very funny. I learned about this first actually in 2022. This kind of this process. So during the Mpox epidemic, I've just been following the literature, what the virologists are working out on the epidemic and so on, and they found out at that point that the viruses from Mpox were mutating much faster than expected than had
Hmm.
been seen before. And it turned out that the types of mutations that they were seeing that were happening rapidly were very similar to this ApoBec kind of known mutation change. And that led scientists to think maybe our enzymes are also working on this Mpox virus. And
Uh,
so learning about
oh, I
that,
see what you're saying.
testing that out, helped to figure out. So we were introducing errors into the Mpox virus very quickly, which made it mutate much faster than usual. And that knowledge helped to figure out better when Mpox actually emerged and when it started to spread.
Oh, interesting. You can somehow work backwards from that information.
Right.
Wow. Okay. Okay. Well, that's a. I am feeling grateful for my immune system has a bunch of tricks I didn't even know about. So thank you, immune system.
I think one more thing is the types of infections and cancers that HIV makes people vulnerable to. I think probably that's not very obvious to people. Maybe they've heard of a few of them. So we talked about Kaposi sarcoma. There are also a bunch of others. There's PCP, which is this fungal lung infection. There's toxoplasmosis, which is this parasitic brain infection.
Right.
There's cytomegalovirus, retinitis that can cause blindness.
Mm.
There's tuberculosis. We already know about tuberculosis, probably. Most people here know about that. That is the leading cause of death in people with HIV worldwide. And
Mm.
the reason is that because of this immune suppression, because of the fact that HIV is depleting our immune cells, it makes us more vulnerable to infections like tuberculosis, which are often easier to clear for people.
Yeah.
And then some of these are also related. So I think I talked about a few that were brain infections or that cause blindness. So some of these can also cause AIDS-related dementias where people lose their ability to think and make decisions and
Yeah.
uh, generally lose their memory. And then there are a bunch of cancers as well that HIV is associated with. So as we talked about earlier, some of our T cells are really important in trying to find potentially cancerous cells and eliminating them. If those T cells are depleted, that makes us more vulnerable to cancers continuing to grow. So that
Yeah.
includes Kaposi sarcoma, which we talked about, and then non-Hodgkin lymphoma, and it also increases the risk of cervical cancer.
Okay, so I think I have a rough understanding of the virus and a rough understanding of how it leads to AIDS and to disease and opportunistic infections. Maybe quickly, where did it come from originally? Where did this virus start out?
So this, I think, has had a lot of research go into it, and the likely answer is that it came from chimpanzees and gorillas in Central Western Africa.
Hmm.
So there are different types of HIV. So as we talked about, HIV mutates very quickly, creates this huge amount of genetic diversity, and that also means that there are multiple different types of HIV. So the
Hmm.
one that's found across the world is called HIV-1. There's also an HIV-2. Both of them are from different kinds of primates, from Central and Western Africa. And I think the current understanding is that HIV-1 came from type of chimpanzee in somewhere near the southeastern area of Cameroon.
Okay.
And probably, so this is interesting. So the way that we understand this is by collecting a lot of samples of HIV from different people, ideally as early as possible. So the earlier the cases are, the easier it is to try to estimate where they all came from or when
Hmm.
they converged back in history. And what was super interesting to me that I was reading about recently was that the earliest genome of HIV was recovered from someone who died in 1966 in Africa.
Wow.
And so
Wow.
that's
Yeah.
15 years before the first cases were reported. So
Yeah.
there were definitely cases of HIV before that, probably for decades before that.
Yeah.
And if you use this sample, but also all of the other early samples that we have, you can kind of trace back where they have shared ancestry in the same way that you might be able to do with a family tree. If you do
Yeah.
that with genomics, you can try to trace back where they have similarities. And that suggests that the pandemic originated in at the turn of the 20th century. So because
100 years
because
ago.
we yeah, well, so we don't have because the earliest case we have is from 1966.
Hmm.
That's still not very early, and that means that there's still some uncertainty in when it
I
actually
see.
originated. So there's this uncertainty between so somewhere between 1881 and 1918 is probably around when it first emerged
Okay. Well, and in humans. So basically, there was
in
this
humans.
virus that infected other primates, chimps, that maybe goes back much longer. I don't know if that's known. And then at some point, someone was probably hunting a chimp, and there was a blood, the blood sort of got into their blood. Or is that the was
Yeah,
it sort of
that's
you're eating uncooked chimp? Oh God.
That's probably it. So probably through hunting primates and consuming them from the kind of butchery of that process and being exposed to their infected body fluids. And so the virus that infects chimpanzees and other monkeys is called simian immunodeficiency virus rather than human.
Hmm.
And
Yes, I am. Okay.
that, it seems like, has crossed over to humans dozens of times.
Oh,
over history.
As in since the
In total.
1900s? Okay, okay. Ah, interesting. I didn't know that.
And I
Okay.
think there's around four of those like spillover events is what they're called. At least four of those spillover events have led to sustained epidemics that
Oh my gosh.
people
Okay.
are still infected
Huh.
by today.
Wow. Okay, so it's been with us for a long time, and we haven't known the extent of it so well as a species until the more recent epidemic starting in the 80s. Is that a fair summary?
That is a fair summary. I think this really reminds me of some of the work and writing I do about missing data.
Mm,
I mean, on a lot of topics in health, but also in other areas, we tend to have much better data collection and understanding of epidemics, but also other diseases in richer countries because of the institutions that can collect that data. So having the resources and the people to collect that data is not
yeah.
super easy. And the fact that we know about the first cases of AIDS that had been reported were in the US is not because it started in the US, it's
Yeah.
because the US had good detection and disease control research going on.
Oh, yeah. I mean, it reminds me of the extremely frustrating initial COVID graphs where reports of number of cases would actually be number of confirmed cases, vastly different than what you could interpolate must be happening in reality. But, whoever gets tested ends up getting reported, and whoever doesn't, doesn't. It's can lead to a lot of missed inferences.
Exactly. That's right.
I feel like we should get to drugs. And when as you've described HIV so far, as a drug developer, which I've never been yet, but moonlight as in my head, I'm thinking the different parts you described are each potential targets that I could maybe make a chemical, a small molecule to interfere with to sort of mess up its life cycle. And I know my immune system's not going to sort it out all on its own. So some of those non-human chemicals might be pretty useful too. So firstly, is that right? And then am I thinking about that right? And maybe that means we're going to have to go in a bit more detail about the infection cycle because the virus is going to look different at different points, and that will maybe give us a clue about what drugs we can make.
No, exactly. I think actually it would be probably easier to think about where scientists were at the time, how
Okay,
much they knew, and how they were developing drugs. And then we can talk about the broader life cycle of the
let's do it.
virus.
Sounds good.
So, place yourself in the 1980s. This is kind of hard because, as a scientist, you had just found out that retroviruses could infect humans. Now you
Mm,
find out that HIV is a retrovirus. You're like, wait, I didn't even know that RNA could be turned into DNA until 10 years ago. So that's quite a tricky position to be in. At this point in the early 1980s, there were no antiviral drugs for retroviruses. Also, antivirals in total were kind of new. I did not know this before reading about it for this episode, but the first antiviral drug of any kind was approved in 1963, which is, again, just
1963. Really? Not even flu? We
around.
didn't have nothing?
Yeah,
That's
no,
crazy.
we didn't. We had flu vaccines, we had a bunch of vaccines before that, but the first antiviral drug was for treating the herpes simplex virus infections of the eye, and that was
Okay.
in 1963.
Wow.
Also, if you were in the early 1980s, you wouldn't have PCR. PCR is polymerase chain
Of
reaction,
course. Yes, of
which is
course.
used to multiply samples of genetic material so you can study it more easily. And that would have been really useful for being able to detect the level of infection, the level of virus in someone. You would be very new to knowing about retroviruses to begin with. You wouldn't have any antivirals for them. You wouldn't have a great idea of how to even develop antivirals at all. You didn't have PCR. You didn't have multi-center trials, so you wouldn't be able to test drugs in multiple hospitals. You wouldn't be able to test whether people were resistant to the virus through the genetics of the virus.
Right.
Again,
Yeah.
because of the lack of PCR testing, and
Yeah.
so there
So
was just this complete lack of what are we gonna do now? We have no idea. This is super new to us. I
yeah.
think a
Yeah.
lot of people at the time might have thought it's impossible to treat this disease. It's so new, it's so different. How are we gonna make any progress on it?
You don't have a proof point you can hang your hat on. That
That's
must have
right.
been very, yeah. The unknown beckons.
And so, again, I was trying to find a retrospective written by someone who works in this field, and I found one that was really interesting by Samuel Broder. He was one of the scientists who developed AZT, the first HIV drug. So
AZT. Yeah.
his team found that it was effective against HIV. They also developed various other drugs against it. And he wrote this retrospective on how they discovered the first antivirals and how pessimistic people were at the time that it was
Mm.
possible to make any treatments against it. And so maybe a bit of background on who he was. Samuel Broder was this cancer and immunology researcher at the National Cancer Institute in the US. And this itself is quite interesting. You're thinking HIV, this is an infectious disease, but the people who were
Mm.
studying it, who made these first effective antivirals were cancer researchers.
So
Um,
he was part of the NIH, the division that works on cancer. And so he was employed by the US government and had a lab in the cancer part. And okay, got it. Keep going.
so I think the reason that he was working on it was, as I said, the first thing that was noticed in AIDS patients that was surprising to people was Kaposi sarcoma, this type of
Right. Yeah.
cancer of the skin. And so he was trying to figure out what was going on here. Um, I read this book about the drug development in HIV and AIDS called How to Survive a Plague. And
Yes.
there they describe this period very humorously to me. I mean, it's obviously not humorous, but the way that they say it was that he was really excited to see this first HIV patient come to the National Cancer Institute. He saw it as this once in a lifetime scientific challenge that brought together two of his interests of immunodeficiency and cancer. And they thought, if we're able to crack this, this is going to be really important. And so I think the first thing they noticed was the virus was probably infecting CD4 T cells. And the reason that they thought this was one of the first signs that you would see in someone who was infected was that their T cell count would drop. So they
Mm.
at least had some blood testing
Right.
and they had some of the tools and technology to measure
Yeah.
CD4 T cells at the time. So they knew that there's this massive drop and they thought maybe it's because the virus is multiplying in these cells.
Seems
And
like
so they,
a link to make. Yeah.
right. So they thought, okay, we've hypothesized that the virus is replicating in these cells. Maybe we should test compounds to interrupt this process, to prevent it from replicating in these CD4 T cells. And so he then approached different pharmaceutical firms to try to get funding to work on this project and also to potentially commercialize a drug if they found one. I think he approached several firms. Most of them said no, but one of them said
Great.
yes. And
Okay.
that company was Burroughs Wellcome. So we probably
Yes,
haven't
which
heard of.
now has a philanthropy, Burroughs Welcome Fund.
Right. And this was a pharmaceutical firm that no longer exists. It was merged into what became GSK or GlaxoSmithKline.
Hmm.
And they were the only company at this time that were willing to consider funding or commercializing HIV drugs. But they were very afraid that their researchers or their scientists would get infected with samples
Mm.
that people were working with. So they refused to work with these live virus samples, and they said,
Oh my God.
"Nope, you've got to work on it yourself." They were saying this to Samuel Broder and his team, and so Samuel Broder's team had to do all of the screening of these drugs, running the trials, all on their own, despite Burr's
Legends.
Welcome then getting the credit for it and then being
Oh
able to
God.
commercialize it.
Well, it reminds
And
me of streptomycin, the
yes,
first TB drug, where there was Albert Schatz, the PhD student, and Waxman. What was his first name? Henry Waxman? Something Waxman.
Selman Waxman.
Salman Waxman. There we go. The professor who then got the Nobel Prize. They didn't share it in the end, did they? I don't know. I'm going to
They
forget the
didn't
story, even
share.
though. Okay,
I
that's
also read
awful.
the story because it was I was writing about antibiotics and it was really interesting because he discovered this group of bacteria that produce antibiotics, right?
Right, in soil. Yeah.
So these it's so weird to think about bacteria that are producing antibiotics, but basically they're producing it to compete with other bacteria. And
Right.
somehow he found that this specific type of or this group of bacteria were producing a lot of antibiotics. And he thought, okay, maybe there's something there. They seem to be killing the other bacteria around them. Maybe we could use that as a treatment for our own bacterial infections. And he started to recruit PhD students to work on that. And one of them
Right.
was Albert Schatz. And what was
And this
really
is the
interesting
Second
about
World War era,
this
right?
was
Yeah.
during the Second World War, well, the 1930s, I think. And he had Albert Schatz start to work on this project in a basement room
Yeah.
that he never, that I think
He's like,
Waxman never
try
visited.
this out on some TB. I'll be upstairs.
And so Schatz was working on this. And then what was really strange was that Schatz was drafted into war.
Oh,
And
yes.
the project basically was on pause for a few months. Apparently, he then got a back injury and then was
Thank
sent back
goodness.
home. And thank goodness he discovered streptomycin.
Wow.
That was the first antibiotic compound that was found from this type of bacteria, which is called actinomycetes. And that group also led to the discovery of various other antibiotics.
Okay, well, Albert Schatz legend. Grad students have been abused for decades, it turns out. And uh, I guess back to HIV, we are currently dealing with, it sounds like, a very virtuous academic group. And, I don't want to insult Burroughs Wellcome, so I'm very glad they brought this to market. So kudos that there was at least one company willing to stand up. So maybe I'll reserve my vitriol in case there are other bad forces I need to get mad at later in the story.
Well, yeah, so this is also not quite as similar because I think Schatz and Waxman then got into this big fight
Right,
over
protract.
who
Yeah,
would be able
I
to
remember
discover
this.
it.
Yeah, had some,
Uh,
and Waxman was withholding royalties from Merck or something. Yeah, there was some.
something like that. But in this case, I think they seem to work together fine. In
Okay,
this case, in Samuel Broder's team, there were a few scientists who were really involved in this. One was Hiroaki Mitsuya, and he was
okay.
doing the day-to-day research on potential drugs that could work against HIV. And I said day to day, but actually they were doing this research in the night after the other colleagues at the National Cancer Institute went home, because apparently they were also afraid
Oh my gosh!
of
Wow,
potentially being contaminated or getting infected.
wow.
And also, there was another scientist called Robert Yarkowen. So these two scientists tested over 180 well, okay, so let's back up to what we said before. You're in the 1980s. You have no idea how to tackle this disease. You have various reasons to doubt whether you can even develop a treatment at all.
Hmm.
And the only clue that you have at this point really is that it infects CD4 T cells. So that probably helps you to test things in a lab. You can probably test how these different drugs affect HIV's ability to infect these CD4 T cells. But you don't really have anything else to go on. So what would you do? And the thing that they did was they just tried anything. They just tried any drug compounds that they had. So Mitsuya tested over 180 different compounds, and they would be coded with different code names. And he would, ask
And
people for whatever they thought might be potentially effective.
there's no unifying hypothesis per se. It's more like ask around, see what people think might work, see what you have lying in the fridge at the Cancer Institute, that kind of thing.
Well, even if you did have hypotheses, many of them would just turn out to not work, and then you would
Yeah,
have to
right.
try something else. And so there were a bunch of hypotheses, but there were also just let's just see what happens. Let's just use this trial and error kind of approach and see if anything works. And
Now I think about high throughput screening, where you screen hundreds of thousands, sometimes more, drug candidates or molecules all at once. Should I be visualizing like where, or is that comes much after, and we're dealing with hundreds?
well, yeah, so I think this might have been before high throughput screening became much more
Yeah,
popular. This
got
was
it.
probably early days where you're doing this one at a time. You have these different
Wow.
cultures of the virus in the lab, and you're just testing out random drugs, one for each one or whatever. Maybe you have a bunch for each one just to see if it's really working. And at one point, they find this one little vial or a bottle with something called Compound S, so that's just the code name. And that somehow seems to keep the infected cells alive. And so this was a real breakthrough. And this compound turned out to be AZT, or azidothymidine. And
Okay.
what was this compound? So this was a compound that already existed, and it was developed in the 1960s, in 1964, by another cancer researcher called Jerome Horwitz. And I was trying to look up who was the discoverer of each important antiviral in HIV, and I found this name. And I thought, okay, let me try to find a retrospective written by him, and I couldn't find one.
Oh,
And the reason was that when he was working on this in the 1960s, he was developing this as a potential cancer drug. So he had this
okay.
idea that if you think about cancers, the thing that people know about cancers is they grow quickly, right? They have these tumors, and the tumors grow quickly, and the way that they do that is by they need to replicate their DNA in order to divide. And so his idea was if you have the DNA code, and the DNA code is duplicated by adding these bases one at a time by our enzymes into this longer DNA structure. What if, instead of a normal base, you had a fake base that was kind of like a normal base, except it didn't allow any more bases to join to it?
Okay.
And so he
Sounds
found
clever.
a compound that it essentially was this type of fake base dideoxynucleoside. And he thought, okay, maybe this is going to stop the cancers from growing. But it turned out it didn't work for cancer. And he was so disappointed with it that he apparently threw away his lab notes,
No.
essentially just trashed it and forgot about it, didn't even apply for a patent. And so it was just in the National Cancer Institute where he also worked, and it was just there as one of the compounds that had been developed.
Don't throw away your lab notes. Don't throw away your lab notes. But I'm glad he didn't throw away the samples. So that sounds great to me. I'm happy.
So we said that the AZT, this new compound, it was able to mimic the bases in our DNA. So why did it work for HIV but not for cancer?
Hmm.
That's something I don't know. But what it does here is almost the same process. So when HIV's reverse transcriptase is turning the RNA into DNA so that it can integrate into our own genome. It introduces this fake base, which
Okay.
blocks the chain from getting longer. It blocks the rest of the DNA from forming, and that halts the virus's replication.
That's epic. Go
It's super
AZT.
interesting. And so yeah, so it was just this I think it's this trial and error approach that sometimes works. What's really useful about things like this is that once you do find a compound that works, you can then try to make modifications that create new related drugs that you now will hope will also work. And that is possible because if there's something about the structure that is allowing it to have this function, then making these different modifications could lead to additional compounds, or maybe it could make it more effective or more safe in some way. And so you could now have this wider range of compounds that you can work with.
Yeah. And my stereotype of AZT is that it was not very safe in the sense of many side effects.
That's right. Yeah. I mean, if I think if you were a patient at the time, you would still see it as much better than the prospects of like a continued progression of HIV.
Yeah.
But it was pretty toxic, so it's affected people's bone marrows, it led to anemia, also just made them feel quite physically weak in some ways. But it did clear out some of the virus from their bodies. It restored their immune function. It cleared infections. One interesting thing I read was that it surprisingly also reversed some of these AIDS-related dementias that I mentioned. So,
Yeah.
because those dementias were actually caused by infections, if you can clear some of the HIV, which is reducing your immune function, which was previously suppressing these infections, if you can replace, you can kind of revert that immune depletion, then you can now fight off these infections that caused brain dysfunction and so on. And
That
so
must have
this
been
was really astonishing, I think, according to Samuel Broder, to the doctors who saw people who were being treated with ACT at the time. They
can't
were
imagine.
genuinely shocked that this was even possible.
And we went from not knowing if any antiviral was going to be possible to you actually seeing as a doctor people reverse even some of the cognitive effect. Yeah, what a time.
What a time. And also, the other downside was not just the side effects, but this drug seemed to work for at least a few months in people, and then they would start to get worse again. And the reason was that HIV would find a way to evade the action of this drug because it was mutating so quickly. It was able
Right.
to find ways to either get rid of this drug or to develop certain changes in its proteins that would mean that the drug was no longer able to work.
Right. So that rapid mutation is that made it so hard for our immune system to operate against HIV now is making it hard for AZT to durably operate against HIV.
But it was really important because it was the first drug. It was a drug against a disease that people thought was untreatable.
Yes,
And this
absolutely.
completely shifted the perception of the disease. Samuel Broder has this line in his retrospective review where he says the question at that point was no longer whether HIV-1 could ever be successfully treated, but rather how fast more therapies could be developed. And their drug AZT moved from research in the lab to drug approval within just 2 years. And this is partly a result of how the trials work, but it's also partly because of activism around trying to make it available quickly.
I mean, that's both so inspiring and so infuriating because so when was AZT available? Did you say in
I think 1987.
1987? Okay, so from 1981 to 1987. If the clock starts at 1981 when HIV was discovered, if there had been more energy earlier, more funding, more support, oh, if it only took 2 years once you started investigating.
It is
Anyway,
really
I should celebrate
frustrating.
the 2 years, but
It is really frustrating because I have been reading this book, the audiobook version of How to Survive a Plague by David French, which is this amazing, very well written book on drug development, how scientists and activists came together to treat AIDS is the tagline, I think. And it starts, I think, in the 1980s, in 1981, and it's
mm,
genuinely so depressing to read, obviously,
yeah,
for several years. This, like, you're getting through this book and you're just so frustrated with how slow people are, how unresponsive, how much they don't treat it as an urgent problem, even when it's clearly an epidemic disease that's growing exponentially over time, that people are just unwilling to consider that there are potential treatments out there, or they're in these petty arguments with each other about what we should be doing. Should we be saying enough? Are we scaring people by telling them that this is a deadly disease? And so on. And it was
yeah.
really frustrating to read. It was a very well written book, but it was very frustrating to read.
I remember it was- I read "And the Band Played On" by Randy Schultz, covering some of the initial years, and had the same experience. Just very, very tough read.
But I think one interesting thing about Samuel Broder and the kind of cancer approach to studying HIV is that I think this. So we said that the reason that they were studying this was because of Kaposi's sarcoma, which was one of the cancers that HIV made people more vulnerable to. But I actually think that being a cancer researcher was probably the right mindset that you needed to have
Hmm.
as a scientist if you wanted to develop drugs against HIV. So one reason for that was cancer research at the time. They were, I think, the only group in the NIH that were experienced with drug development. But the other was just you're facing this horrible disease that's very rapidly progressing, similar to cancer. You're also in the situation where action is much more important than inaction because it's just going to get worse. You're also in the situation where you're willing to take drugs that have toxic side effects, even if that
Right.
because
I
they
mean, yeah.
might be able to slow down the disease, and that's more important right now because the disease progression is so deadly. But I think the next thing is because you would realize that what was really important here was not just using a single drug. So in just like with cancer, just like the connection to Jerome Horwitz, who discovered ACT, who was also a cancer researcher, you would know that cancer and HIV were rapidly able to evolve to mutate and develop resistance against any drug that you developed. And so the aim would not be to develop a single drug, but to use a combination of drugs. And that was the goal that these researchers had even in the 1980s. Even though they developed AZT, it did work, but people eventually started to develop resistance against it. But that was okay from their perspective because they knew that this was not the end goal. You
This
didn't
is the
just have to
beginning.
develop one drug, we had to develop many.
We have to develop many, not just because they're going to get used one by one and then become resistant, but to use in combination from the beginning. So they were thinking that way from the beginning. That's, yeah, okay.
Yeah, I mean, this was so interesting to me, just as how to develop drugs. What is the mindset that's required? What is the type of approach you use of just trying everything, essentially having different hypotheses, just seeing what works. I thought it was really interesting to read about. And yeah, so this, I think, then spurred a lot of other pharmaceutical firms and researchers to work in the area to develop other types of drugs. Samuel Broder's team then developed a bunch of other similar drugs. So we just talked about AZT, which is a type of nucleoside reverse transcriptase inhibitor, NRTI.
Hmm.
And as we said, it's a drug that is this mimic or this fake version of a nucleoside base off the DNA molecule of HIV.
Let's pause there and let me see if I can remember everything I just learned. So I'm putting myself in the headspace of a drug developer who doesn't have the tools of 2025 when we're recording today. And those are quite a few tools I don't have. I don't have PCR. I don't have, and I don't have modern genomics. I probably don't have high throughput screening. I definitely don't have knowledge of what HIV looks like in terms of a visually as a 3D structure. That's probably far away. And I don't really know the whole process of the life cycle of the virus. But what I do know is that probably CD4 T cells are implicated because I'm seeing these counts drop. I'm taking blood samples and those counts are not looking so good for patients. And what I do know is that if I rummage around, there are going to be some failed cancer drugs somewhere I can at least try. And so, sure enough, because, and in addition, if I'm a cancer researcher, I think about resistance and I think about combination drugs. So what I'm going to do is I'm going to go around and try a bunch of stuff. I mean, my takeaway from this is it was just incredibly empirical. You didn't have much in the way of theory beyond the CD4, sort of implication, link, and you were trying stuff. And a bunch of stuff probably did not work. And then guess what? One thing did work, and that gave everyone some hope and changed things going forward.
Yeah, I mean it's so amazing to read about drug development during that time and what happened after that as well. So maybe this is the time to actually talk about the HIV life cycle and
Okay.
what the other types of drugs that have been developed are. And so we should start with how an infection happens at a molecular level.
Great.
So we have the HIV virus particle. I don't know if people have seen an image or diagram or something of HIV, but essentially it has this.
I'm holding up my hands for people watching the video. Does this look right?
That looks right. So this is a spherical particle. Has a bunch of proteins coming out of it. And inside the spherical particle is a bunch of stuff, including capsid. And this capsid is the core of the HIV virus. You can think of it as looking, oh, wow. Is that an
I just
actual...
picked up something to mislead people. It's a sun-bleached version of a vaccine, but that mimics a virus's structure so that you can present to the immune system. This is a COVID vaccine that the Institute for Protein Design made.
Wait, can you hold it up to the camera?
Yeah, oh yes, I'm looking at it for myself. So anyone watching the video here, this looks like a virus. It is not a virus, and it presents the receptor binding domain of the spike protein of COVID or of SARS-CoV-2 to the immune system on lots of different places, so you can get antibodies that bind it. Doesn't look that far off HIV. I mean, it's better than when I held my hands up. So
it looks kind of cute also.
It is cute. It is cute.
I had a stuffed toy version of the coronavirus that I got from this museum. And I thought it would be really funny to get this as a gift and then to give it to someone and say, ha, I've given you COVID.
It's crucial to get good bits in. So I support.
So the HIV virus, you showed the spherical particle. So we have this envelope that is a sphere, and then it has some protein sticking out of it. Inside it, it has a capsid. The capsid sort of looks like a thimble, or maybe more like a bullet. And this is kind of interesting because the bullet or the capsid contains a bunch of the really important stuff for the virus. It contains the RNA molecules, that's its genetic code. It also contains a bunch of other enzymes that it needs to do important stuff, including reverse transcriptase, which it needs to turn its RNA into DNA, and a bunch of other enzymes that we'll come to. So we have this HIV virus particle, the spherical thing with the protein sticking out of it. One of those proteins is called GP120, and that protein when the virus gets into our body, it targets our white blood cells, our T cells primarily, and this GP120 protein attaches to a CD4 receptor on our T cells.
Got it.
They get attached.
So the virus is
Then,
currently outside of the cell and it attaches to
right.
CD4 on the outside.
Then it starts to also attach to another protein, CCR5, and or CXCR4. There's like a so initially it starts by infecting CCR5 T cells. So it uses these two receptors, it binds to these two receptors, and then it injects itself into our cells.
So it binds to two parts on the outside of the cell, uses that as a way to get inside.
Yeah, so it fuses with our cell membranes,
Yeah.
inserts the contents of this HIV virus into our cells. That includes the capsid, the bullet-like
The bullet.
thing. But I think a really good analogy maybe is like a rocket. You know how when a
Oh.
rocket launches, most of it falls off, but there's this core part of the rocket that continues going.
Right. And usually that's something I like because it contains astronauts. In this case,
It's not.
I don't like it. I don't like it. So,
So
instead of space, we're now in the cytoplasm.
That's right. So we're now in the cytoplasm, the inside of the cell. At this point, the capsid then makes its way to our cell's nucleus. This was
Where the
really
DNA
interesting
is.
because so I had watched this video to try to understand what was going on, what was the pathway. I feel like videos kind of help me remember things better. This video was from 2010, I think. And then I started reading about this process separately in research papers, and they described it differently. And it turned out that our understanding of this life cycle has actually changed in the last 5 years, right?
Yes, I actually talked to a friend who did her PhD, who I think graduated in 2018 and did her PhD on the HIV capsid. And she was saying to me, oh, back when we were doing it back in 2018, all those centuries ago, we actually didn't yet know that the capsid, at least sometimes, makes it all the way intact into the nucleus. I was like, what? But that's so basic. That's the whole game. And it turns out, no, even now we are getting tools that are making it easier to actually see what the heck is going on inside these incredibly busy cells. And it makes you wonder if you do a PhD in 5 years, what the heck are we going to know now?
I mean, I think one of the reasons for this is that it's really hard to observe an infection happening. It's obviously very harmful and probably very unethical to infect someone directly with HIV if you wanted to study what happens in this early part. And so the people with HIV that have been part of research have obviously been much further along than just being infected. And so it's like hard to actually study those earliest stages. And that's especially true because HIV doesn't infect other animals. The closest that we could use is SIV, simian immunodeficiency virus, which is slightly different. And that means that there are various things about this early stage that I think weren't very clear. And I think that the change in the last few years was better microscopy.
That is what my friend was basically saying. She was saying, before we had cryo electron microscopy, cryo EM, we just couldn't visualize things as well. She was using other techniques to do her best, and now you have this atomic resolution of these systems that we haven't ever seen. I mean, it's beautiful. You can actually see what's happening. We never knew. Yeah.
It's genuinely crazy. I mean, I've been reading about sort of vaccine development, biology over the 20th century, and what is really surprising to me is that we didn't have any way to visualize viruses until the 1930s. Before that, so we have the smallpox vaccine, which is against a virus, but this is before anyone knows what viruses are. That's before germ theory was developed. We just happened to get quite lucky with observation
Yeah.
and,
Right.
testing. But it's only in the 1930s that we actually got this ability, this type of new microscopy technique called electron microscopy that allowed us to see things at the resolution that would let us see viruses that are much smaller than other bacteria,
Yeah.
parasites, and so on.
Okay, we're getting too excited and we need to focus on the life
Oh,
cycle. I
that's
can
right.
tell, I can tell, I can tell. Because right now I'm a capsid and I'm about to enter the nucleus, or I'm in the nucleus. What's happening after
Okay,
that?
so let's recap. So the virus attaches to the cell with GP120, attaches to CD4 and CCR5, inserts itself, fuses with our cell membrane, inserts its content into our cells. That includes the rocket or the rocket
Rocket.
core or the capsid or the bullet, whatever. That capsid makes its way to our cell's nucleus.
Mm-hmm.
It then actually gets inside the nucleus, and so our nucleus has these entry points, which are called pores,
Mm.
and the capsid kind of snuggles through, kind of wiggles through those. And this is going to be important later on, the wiggling. And so it gets into the nucleus, and then it starts reverse transcription. So at this
Question,
point,
question,
oh,
question. Does it do another? So I came as a full package and then I unfurled myself for the inner package. Do I unfurl myself again so the capsid kind of lets all the inner contents out into the nucleus?
I think so. And I'm afraid of saying anything too definitively because I'm thinking, what if this knowledge changes in a few years or something like that? But I think the capsid also dissolves at this point.
Okay.
Within the capsid, we said that there was the RNA, the two RNA molecules, and there's the enzymes, including reverse transcriptase. So reverse transcriptase turns the RNA molecule into DNA.
Mm-hmm.
Then, now it has DNA. We also have DNA. It can then insert itself into our
Yeah.
cell's DNA
Yeah.
using an enzyme called integrase, which integrates it.
Integrate. Yep.
Makes sense. So the virus has now integrated itself into our cell's DNA. Now, at some point, our cells will decide to turn our DNA into other RNA molecules and to proteins because we need parts of our genetic code to do stuff at different
I don't want to
times.
brag, but I'm doing that all the time
We're doing that
right
all
now.
the time. I don't know the maths on this, but I know there's a lot of it going on at any given time. And so it basically uses our own cell's machinery to
Right.
turn its DNA now into its RNA particles and also to transcribe the other proteins and enzymes that it needs for its functions. These proteins and enzymes and the RNA molecule somehow make their way to the surface of our cells. They then bud out of the cell, so the envelope, the cell membrane of our cell, which previously was fused with this with the previous virus particle,
Mm-hmm.
they bud into this new little particle, and there's now an immature virus, new HIV virus particle. But there's lots of them because it's not just that our body has transcribed one of these. It's transcribing loads of these
It's doing
proteins and
loads of them.
enzymes.
Yeah.
So this one HIV that has infected our cells and turned and integrated into our DNA can then multiply into many more that bud
Right.
out of the cell. But at this point, it's still immature. It's still not able to cause an infection because it hasn't the proteins that we've made for the virus are actually in this big compound of this what is called a polyprotein. So it's multiple proteins that are fused together
Mm.
at this point. So
And so they're not
the
that it was maybe more efficient for the virus to do them all at once, but they're now in a big string, so you can't actually they aren't going to perform their function. There are many different proteins that it wants to use. For example, the integrase. Maybe it's still in part of that. Is that right?
They're all part of, well,
Okay.
no, I think there are multiple polyproteins, but
Okay.
one
Okay.
really big one has reverse transcriptase, integrase, and like a bunch of other important
Got it.
proteins. And they're all kind of in this huge polyprotein. And then there's a separate enzyme that's produced called protease in HIV.
That's what I would do. If I had something that I think I know what's going to happen. Keep going.
Okay, so this protease is what is commonly called a molecular scissor. It
Right.
cuts this giant polyprotein into its components, into the individual enzymes that it then needs. And that also that also cuts off capsid proteins, which then form a new capsid.
Oh, wow. Sorry, we're inside of the envelope right now of the virus,
We are now
but
inside
there's
the
just a lot
envelope.
of mess in the immature
Right.
state. We
There's
don't
just
have a capsid yet. Okay.
so we now have the protease has cut all of this giant protein, has cut them into lots of capsid proteins. The capsid proteins start to assemble into new capsid.
Which in itself, I mean, that's so cool because there are loads of different proteins, right? That's going to be lots of different individual units that are sort of due to, thermodynamics, I guess, sort of gravitating towards this configuration that is a bullet or it's
Yeah,
wild.
I mean it's crazy. And so this protease is what I wanted to talk about a little more. And I think it's probably really useful to hear about this whole life cycle in order to know what protease even is. And so the HIV's protease, which is cutting up this giant protein into its components, that is what I think is the next big advance in HIV drug development. So there are a bunch of other nucleoside analogs, the mimics like AZT, that are developed around this time. A bunch of other drugs that are developed, but I think the proteases are the next big step. All right, so we talked about protease, and the reason that's important is because the next big advance, in my view, is drugs that targeted HIV's protease enzyme. And so I think I'll talk a little bit about the first one that was developed. This was called sequinavir, and it was developed by scientists at Roche. And they were trying to figure out if there were any drugs that could target this protease. So they knew that this was probably important because they could see that I think they were able to test whether it was present in people with HIV. And they knew that it was important in the process of breaking down that giant protein polypeptide into the smaller components. And so they started to study its structure and its cutting pattern. So as we said, protease is often called a molecular scissor. It doesn't literally look like a scissor. I assume it just looks like a blob or something. But when it's when it's trying to cut down this giant polyprotein, it slightly changes shape. I guess it opens up. It gets into this transition state, attaches to the polyprotein, and then snips it into separate proteins. And what they were
Right.
trying to do was they're trying to find something that could jam that transition state so it
Mm,
couldn't actually cut the protein.
right.
So you have to look at what this transition state might be, what specific part of the protease enzyme is doing that snipping, and then can we fit something into this little gap?
Okay, so we've got this protease scissors, and we got my long string of proteins paper, and we're going to jam a rock in there. And rock beats scissors. And that's one thing I learned many years ago.
Very good.
Thank you.
So, what was interesting, I think, to them, I think at this point there is PCR testing because this is 1986, so now PCR is available. but they could also try to look at where exactly the protease was typically cutting. And they found that if you looked at protease in other proteins in the lab, it was cutting at specific sequences in a protein. So a protein is made of many amino acids joined together, and it was typically cutting in places with a tyrosine, which is one type of amino acid, or
Hmm.
a phenylalanine, which is another, and
Okay.
either of those followed by a proline. And so it was a sequence of either tyrosine or phenylalanine followed by proline. And this combination of cutting at this point is something that human enzymes almost never do, which is really useful because it means that if they're able to target something that is cutting at this point, then they're hopefully not going to be affecting any human enzymes
Yeah.
that are important to us for our other functions.
Right.
So, what they then did was try to look for other molecules that could fit into this transition state where the enzyme is snipping the
Hmm.
polyprotein. So, as part of designing the first protease drug, they also had to develop tools to test how well their drugs were working against it. So they developed a dye reaction test to detect these proline-containing fragments. They also worked on cloning and purifying the protease enzyme using recombinant DNA methods, which are also fairly recent. The first recombinant DNA that was produced was in 1972, and the first time that it was used for human enzymes was insulin in 1978. So this was the first time
Right.
that we could produce insulin in bacteria instead of extracting it from the
Pigs,
pancreases
was it? Who was? Yeah.
of, I think, pigs. Previously to that, it was dogs, and it was also
Oh,
cows and other mammals, which is horrible. But like that,
yeah.
there was no other way to treat diabetes except to extract insulin from various animals until the 1970s. So this was a huge development that was also very useful for testing out potential drugs against protease.
I mean, yeah, we now take this for granted. In any lab you're in, or most labs you're in, you'll have some way of growing up proteins you want to study in a biological system, in a probably in a bacteria, but maybe in mammalian cells, maybe in some in yeast, and that means you can study proteins all day long. But back then, this would have been recent. You know, you could only do that from the 70s, as you said. So yeah, that's another nice intersecting biotech improvement there.
It's so interesting to me just how much the technology kind of happens alongside and how much is
Mm,
dependent on other tools being available and
yeah.
what new things that allow scientists to do. Okay, so back to protease inhibitors. We're trying to test any drugs that fit into the transition state, the little wedge where the scissors are cutting. They tried a bunch of different drugs. One type called hydroxyethylamines worked especially well. And when they found that that was working, they started making adjustments to it to see if they could improve on that result. They changed the ends of the molecules, they tweaked the sizes of chemical rings, they swapped side chains, and they found that having a larger fused ring structure made it much easier for this compound to latch onto the protease and block it from cutting.
I find this kind of stuff crazy just because the tweaking is so important. And we're talking about very small molecule. I mean, well, small areas we're tweaking. We're talking about around that many atoms. We're talking about, you add up all the atoms at the cleavage site. I don't know, not that many. So yeah, it's wild that chemistry gets so specific, and that means that you can have these small changes that have enormous changes for patients.
Right. I mean, I think it shows how much different fields of research come together in developing new drugs. We have people who work on in the clinic on with patients, and they might see something seems to be having an effect, so they try it out. Then there's the people working on microscopy who are really important, the chemists, the pharmacologists who are testing out toxicity and drug reactions and things like that. It's like everything comes together
Yeah.
is really important here. And so this drug that they then developed after these dozens of adjustments was initially called Row 31
Row 31.
8959.
Okay,
That
row
was later
31, 859.
named
That rolls off the tongue.
that was later named Sequinavir, which is the first protease inhibitor that was approved. And as I said, this drug, because it was targeting this the enzyme that was cutting in the specific place that was not the case in for human enzymes. It was extremely selective to HIV protease and barely affected human enzymes, even at very high concentrations. And that meant that it was much safer. But at the same time, what I found kind of interesting was that this drug, if you gave it to people, most of the drug was excreted very quickly. About 96%, I think, was excreted in the urine.
Hmm.
So having a high concentration of the drug thankfully didn't have these side effects because you'd
Oh,
have to
wow.
compensate for the amount that just gets peed
Huh.
out.
Wow.
And I think this is also interesting because we're at this point now. This was approved in 1995, Sequinavir. And at this point, there are, I think, around a dozen different antiviral drugs, which again is amazing because just 10 years before that, people thought no drugs would work against this disease, but now they have 10 or so.
Progress.
And so at this point, there are a bunch of drugs, but none of them really work in a long-lasting way. HIV manages to evolve resistance against the drugs that are being used after a few months. And this seems like it's just another one of those, okay, we've got a new drug, but is it really going to make a difference in the long term?
Mm-hmm.
And I think people were in I think in terms of people with HIV, they were probably quite pessimistic in some ways. They want new drugs to be developed for the condition, but how is this going to make a difference
Yeah.
after a few months? But this is actually where things change. Because now that we have a protease inhibitor and we have the nucleoside, the fake nucleoside state like AZT, and we have a few other drugs, we can now combine them and give them as combination treatment.
Ding, ding, ding. Here we go. And I don't know if literally the combination involved AZT or some of the other antivirals you mentioned, but I did notice that, the protease is operating right near the end of the viral life cycle. The AZT, as you described, it's operating much earlier. So these are really different parts of the life cycle. And, it's quite unlikely intuitively the virus would at the same time mutate against both, right?
Right. And so I think this is something that the cancer researchers or the virologists working on this would now have realized that we're now working on different aspects of the virus's life cycle. It's fairly unlikely that it's going to be able to resist all of these drugs coming at it in this different parts. And so I think this is when this combination therapy started to be used, and it was being tested in alongside these new protease inhibitors. And this new type of combination therapy is called HART, which stands for Highly Active Antiretroviral Therapy. And it's a combination of typically one nucleoside reverse transcriptase inhibitor like AZT, another drug inhibitor that directly inhibits reverse transcriptase, so it's not a fake base, and then a protease inhibitor. And there were multiple protease inhibitors that were introduced around the same time in 1995.
Okay.
Like a bunch of different pharmaceutical firms essentially racing to get theirs to market. And this is a huge change in terms of how HIV treatment works, how effective it is in the US. And I'm showing this chart that I worked on earlier just to show what impact
Oh
it had.
my
So you can
God.
see this massive rise in mortality rates from HIV and AIDS from the 1980s to the 1990s, rapidly grows as an epidemic disease, growing exponentially. And in 1995, in December, highly active antiretroviral therapy, the combination treatment, is introduced, and it's just this huge drop in death rates. And the way that people talk about it is as if people are on death row and they're coming back to life suddenly with this new combination therapy, because it's something that the virus is very hard, not able to evolve resistance to. And I wanted to bring this up because I was reading that book, How to Survive a Plague by David France. And he talks about his own. He was a reporter at the time, and he was at one of these scientific conferences on protease inhibitors, learning about the science, what new drugs were available. He describes one of his experiences towards the end of the book, and this is what he says. He says one of the scientists interrupted his presentation abruptly. And he said, "Maybe you are not understanding what I am saying. This is the biggest news ever in this epidemic. This stuff is actually clearing virus out of people's bodies. People are getting better. We don't know for sure yet, but we think these drugs, this whole class of drugs, might allow people to live a normal life. This is what we've been working for all these years. They're not a cure. We don't know what they are in effect. But this is the first major piece of good news we've had in all these years. They're calling it the Lazarus effect. People who were in hospitals on their last breath are getting up and going back to work. We've never seen anything like it." And that's just an incredible change, I think, from how scary it must have been in the 1980s. Seeing some drugs really promising, but eventually they start to fail. And then you get this combination therapy that changes everything.
A complete change. And it breaks your heart to think of people who didn't make it to see that change. Oh, what a graph. Okay. You're going to have to make you have to make some nice, less emotionally intense graphs for me to calm down now.
Yeah. So there are a bunch of other drugs that I think we aren't going to talk about, but apart from the protease inhibitors, the nucleoside analogs, and other reverse transcriptase inhibitors, there are other drugs that target how HIV enters the cell. I think there are also some that target the integrase enzyme that allows its DNA to integrate into our DNA. And then there are some others as well. But I think that kind of covers much of the major story in the 1980s and 90s on HIV and drug development.
Okay, so taking a step back, we know what HIV looks like. We know roughly where it came from. We now know a bit about the life cycle, and we know about combination drugs that together prevent HIV from taking over and are less prone to resistance because as many of them you're on at once. Okay. What about what's happening with vaccines at this point? And are people talking about curing HIV? I mean, these drugs control HIV.
I think this is interesting because we don't have a vaccine for HIV yet, right? It's been more than 40 years since the first AIDS case was reported. We have loads of antivirals now, working in different ways, but we don't have any vaccines. And I think this would have been really depressing if vaccines were the only things that were being worked on at the time. But thankfully, people were trying out random drug combinations. And that's why I think this first step of getting ACT was so important.
Yeah.
But my understanding is there aren't any working vaccines that we know of yet. So I didn't actually read anything about them.
Well, I know a little smidgen. So at Open Philanthropy, where I work, the team I work on supports a bunch of different biomedical research, and roughly a third of what we fund in grants is in some way or other vaccinology or focused on vaccines. And the area of vaccines we've done maybe the least in is HIV. And the reason for that is that it's very hard. And now, people have more knowledge of why it's hard. It also happily, despite being so underfunded at the beginning of the crisis in the US and elsewhere, now has attracted much more attention and funding. So there's actually been tens of billions of dollars thrown at this problem of how do you make an HIV vaccine? The reason it's so difficult, I think the clues to that are located in what you've already said about the virus. So, well, firstly, the clue number one is that our immune system doesn't control the virus naturally very well once an infection is established. So most people are not able to control an infection once it's established, and that implies, okay, well, what is a vaccine? A vaccine is trying to trigger your immune system to be prepared for future invaders. If hardly anyone has a prepared, successful immune response, what are we even trying to mimic here? It's tough problem statement. Whereas something like COVID, plenty of people do manage to control, and their immune response is productive. And you can pretty easily see with COVID, especially at the beginning, well, there's one protein on the outside of this virus, the spike protein, that if you block it with antibodies, it is not getting into your cell. So let's try and mimic that immune response. And then another clue is about the rapid mutation. So if your immune system is trying to prevent something that keeps changing, it's going to be harder. And sure enough, if you create a vaccine which is less dynamic than your immune system and is only kind of one thing, then you're not going to be clearing all of these different, permutations of the virus. These days, because the understanding of the immune system has progressed even outside of HIV over the last few decades, and because we have so much better tools, people are still going at the problem and have sort of ingenious and complicated ideas about how to make a HIV vaccine. You may have heard the phrase broadly neutralizing antibodies. That's all the rage for what people are using to develop vaccines and going after. But that's kind of importantly not what we're here to discuss today, because I think you and I really are focused on medical impact, and this podcast is too. And it's so interesting that what we are here to discuss is a drug in the sense of it's not prompting your immune system to respond in a certain way like a vaccine would, and it's trying to avoid getting rejected by your immune system, and instead is trying to just be a chemical that's hanging around, and the chemical's doing the work, not your immune system.
I was thinking about the broadly neutralizing antibodies. So just in case people are not aware, this is I guess a fraction of people seem to be able to develop an immune response to a wider range of HIV strains after it's diverged. And so trying to find those antibodies that seem to be working against this broad range, that is what people are looking for, right?
Right.
And the other thing is there are some people who are working on combination vaccines, I think. So vaccines that include multiple different components of the HIV virus. And this I know from direct experience because I was once in an HIV vaccine trial.
Aha! So when was this?
This was in 2019. So
2019.
you might be wondering, why am I getting an HIV vaccine? Why am I in this trial? It was this phase 1 trial. So essentially, they're just testing the safety and some basic reactions, immunological reactions you have to a potential vaccine. And I got contacted through Imperial, where I think I was studying time or had been.
Okay.
And I was like, I love science. I want to be part of this trial.
We need more Salonis in this world.
And also, I was thinking, well, what if the vaccine, what if this candidate vaccine actually works? I'll be immune to HIV. That would be so cool.
Well, oh yeah, so this was not controlled because it was a phase one, so you were definitely getting it.
Oh, I actually don't know. I think I
Okay.
mean, I could have been on placebo, but still, you have a 50%-ish
Take the
chance
50.
probably
Get it for
of
free.
getting an HIV vaccine for free if it works. And it was a really funny experience because if you have met me or seen me in person, I'm quite small. I thought that I knew that this trial had this eligibility requirements that you had to be within normal BMI. I am like essentially on the cutoff of underweight and normal whenever I've checked, and that's just been true for years. And so I was really worried that I would just fall under the threshold and I would not be allowed to participate in the trial.
I love that you're hustling to get into an HIV vaccine trial. You're like,
I'm like
no,
eating
don't
more
screen me
food to
out.
get in. Exactly.
Bulk season is on.
So, okay, so I was really hoping that I would get into this trial. I got to the clinical trial site. They asked me a few questions. They asked for my consent, etc. And then they also wanted to measure me to check that I met the requirements. So they were
Oh no.
measuring my weight and my height. They then put that into their computer and they said, "Oh, great, you've passed the threshold." And I saw this BMI value on their screen. I was like, "That's surprising. Great, but surprising." And then I looked at the values that they had entered, and it turned out that I was shorter than I thought I was. So my BMI was normal.
So it took medical development of HIV to get you to understand your height. I mean, there's a lesson here, but I'm not quite sure what the lesson is.
Well, it's hard to measure your own height. And
Great point.
I don't know. I, it was both exciting because I could now participate in this trial, but also there was this sadness that I felt through realizing that I was even shorter than I thought I was.
Oh god. Wait, so what happened? Did you are you protected?
I don't know. Well, they didn't unblind me from whether I was getting the vaccine or the placebo, but I did go in. I think I was in for some 8 sessions. They did a
Oh,
bunch
okay.
of, was it blood testing? They did some testing of uncomfortable parts of my body to see the effects of this vaccine. I don't think I had any side effects, maybe a
Okay.
headache at some point, but that was all. Um, it was pretty nice. It was a great experience.
Nice. Cool.
I would, I mean, I would say I would recommend it, but really you should decide that for yourself.
Sounds good. I just recently was screening to sign up for a vaccine trial at a here in San Francisco, and I did the 15-minute screen, and they signed me up to go in person. And then the day I was going to go in person, I had a meeting that clashed, and I haven't got around to enrolling, so I'm feeling a lot of guilt.
Oh.
So now yeah, now I have an extra incentive though. Maybe I'll figure out I'm taller than I think I am.
Mm.
Maybe I'll figure out I'm shorter though. So
Well, so the reason I brought this up was because it was a combination vaccine, but also it was a funny story. But
okay.
the vaccine that they were trying contained, I think, 3 different proteins of the HIV. So I think it was one adenovirus that was modified to carry an HIV coat protein. There was another that was a vaccinia virus, which is that the smallpox vaccine virus? I think.
Yeah, probably.
And then there was another that was another coat protein. So they had tried out, I think, one or two of these before in trials, and then this was putting them together into this combination vaccine.
And it's
I don't know
interesting
how it
that,
worked out.
well, yeah, it's interesting you mentioned a co-protein. So what, it makes me think of the design differences you're dealing with when you're trying to make therapeutics and when you're trying to make vaccines. And with vaccines, stereotypically, especially for antibody responses, you want to look on what's on the outside of an invader. What's sticking out that my antibodies can glue to. And maybe a coat protein's a good choice because it might be sticking out. And you know what is not sticking out? Those strands of RNA that are not only
Right.
inside the envelope, they're inside a capsid. Your antibody is not getting in there.
Yeah.
However, a small molecule drug, which is a nice tiny little chemical, can diffuse to many places very surreptitiously. And so you really might be able to interfere with something that the virus has tried to protect from your immune system, but has failed to protect from genius humans who are using good tools to make something that nature actually couldn't have really got to.
No, exactly. Yeah, that's a really good point. Shall we talk a little bit about treatment for HIV and what that's like?
Sounds great. And maybe we should even skip to prevention. We've talked a bit
Let's
about,
do that.
you know, let's do it because you've given us a good overview of how in the 90s these new drugs allowed patients who had HIV infections to have much longer life expectancy and control their infections. And, there's a lot more that we could say about the different improvements since then in treatment. But, the principle is somewhat similar if you want to be on these combination therapies. So let's skip to prevention because prevention has some overlapping path and some different path. So, what were the first ways that you could try and prevent getting HIV if you didn't have it yet? Well, you could change the way you were having sex, the type of sex you were having. You could have sex with fewer partners, and you could have sex with condoms, which provide a barrier. The thing that really changed preventive strategies more recently, though, was drug availability for PrEP, pre-exposure prophylaxis. So that's different than post-exposure prophylaxis, which is PEP. And the pre means you're taking the drug before you have sex or before you get exposed in some other way. And that means that if any HIV particles enter your system, the drug is going to help block an infection getting established. So PrEP as a drug regimen first became available in the US at least in 2012. So Truvada is a combination of two drugs, tenofovir and emtricitabine. Do you know how to say that one?
No.
Okay. They put them in one daily oral pill. So more specifically, it's actually tenofovir disoproxil fumarate, which I'm sure I'm also mispronouncing, or TDF, in combination with emtricitabine. And so those were two separate drugs that had been approved for treatment of HIV in 2001 and 2003. The combination of them was approved as a treatment called Truvada in 2004. And then by 2012, the FDA approved Truvada as the first PrEP regimen after a clinical trial showed that it had high efficacy in preventing infection.
It's so interesting that some of the same antivirals that are used in treatment were also used in prevention. One
Yeah.
thing that made me think about was I was reading about azidothymidine, AZT, the first HIV drug. And I think there's a part of that story that gave them a clue that that antivirals could be used as prevention as well.
Okay.
And that was that pregnant women who had HIV, who were taking ACT were not passing it on to their babies at the same rates. And so they started running
Very
this
interesting.
trial in the 90s. And in 1994, I think the study was published. It was this massive drop in the rates of transmission from mother to child of HIV.
Wow.
And that is really interesting as well, because even though people were developing resistance to different HIV drugs, if they were pregnant and taking it, the drug resistance was not as much of a problem
Hmm.
if they were taking it late enough, because you only need this particular time span for it to be effective. It doesn't have to be effective for years or more
Ah,
than
of course, I
months,
see.
right?
Huh.
But also, I think that just gave people a hint that this is something that could be used in prevention.
That's such a neat real world kind of proof of concept of what you can do there. Truvada has been improved on since, so maybe I'll just go through a couple of those improvements. The fundamental idea is similar for the main improvement drug. So in 2019, there was a new regimen called Descovy. You might be wondering, is this from a competitor who's trying to outdo Truvada? And it's from the same company, Gilead, who, as a bit of a spoiler, developed Lenacapavir later in life. And Descovy does have a longer patent though, so it's a better variation for men. It's emtricitabine again, which I've probably said 3 times in 3 different ways now. It's tenofovir again. So it's the same dose actually of emtricitabine. I think it's 200 milligrams. The tenofovir is in a new form though. So it's instead of TDF, it's TAF, which stands for tenofovir alafenamide. And both TDF and TAF are prodrugs for tenofovir. That means your body is sort of doing some work once you ingest them to enzymatically convert them into tenofovir and then into tenofovir diphosphate, which is the active form of the drug. And my understanding of the difference is that for TDF, so the original one, that primarily happens in blood plasma, and for
What
TAF,
happens? The
the
change,
conversion into
right?
the active drug, and for TAF, that primarily happens in the immune cells. And if you think about the difference there, well, getting the same thing out the other end, why do I care? Well, if you're doing it in the blood, then your blood's circulating everywhere, including your kidneys, and you can actually have more unwanted effects from that than if you are more secluded when you're
Ah.
making your active drug. So I think that's why the safety profile of Descovy looks a little bit better. So you have, if you're on long-term daily use of the first one, it's got a pretty good safety profile, but it can have negative effects on kidneys and bones, so bone density and kidney toxicity. And so here's where if anyone's on the video, I'm going to do some show and tell. So I don't know if are you the kind of person I keep all of my empty pill bottles into the future indefinitely.
Oh, I
Yeah.
I don't do that.
So I
So
do
these are
this because
empty pill bottles
these are
on.
empty pill bottles, which hopefully don't have private information on them.
I think.
But basically,
Ooh.
I think I do it because I have some vision of I'm going to do some art project about
Oh
what
wow.
it's like to be a modern human in the future, and you do that. But I think I
You
actually
should do that.
stole that from. I think I've seen an art project which is loads of empty pill bottles, so I actually don't have a plan for these pill bottles. But basically, here's what you can learn. So take one tablet by mouth every day. This is citrabine and tenofovir,
So cool.
200 to 300 milligrams. Here's another thing you can learn. So this empty pill bottle says Loris Labs on it.
Hmm.
And you might be thinking, Loris Labs? That doesn't sound like Gilead. True. And this other empty pill bottle says Amneal on it. Doesn't sound like Gilead either. And the reason for that is that the Truvada patent expired in 2020. So there are now many generic drug manufacturers who make Truvada, which is why I am on Truvada, because when I first asked to go on Descovy, my doctor at the time was like, I think that patent expires in 2031, was like, No. We're going to give you Truvada. And then I did the, classic.
Because it's cheaper.
Yes,
Okay.
correct. So it's made by um, drug companies in outside of the US, usually in middle income countries or in lower income countries, a lot based in India, but other countries too. I actually don't, actually, I feel like one of these is an American company, but I might be wrong. And by sort of making these generic competitors where they only have to prove to the FDA that it is similar enough in terms of its, pharmacodynamics and all that, they don't have to redo all the clinical trials. They can sell for a cheaper price than Gilead might. I do remember later on, the first injectable PrEP came about, so cabotegravir. And that is made by a different company called Viv, VIIV. And I believe that was approved in 2021, if I'm remembering right. And that is, you only get one injection every 2 months. And so I had heard about it and asked a different doctor I had at the time, could I, just asked about it, wasn't sure if I'd do it, wasn't sure if I'd stick with what I was doing. And that was very expensive. So that was a quick no. My insurance at that time was not enthused about that one. If memory serves, it was roughly $4,000 a dose. And
Oh,
there's,
wow.
it's every 2 months, that's 6 doses a year. I think it was roughly $20,000 a year. And the patient benefit, sure enough, was not, I shouldn't really have been paying that much because the drugs I was on were working perfectly fine.
I guess I'm interested about how prep works on a day-to-day basis. Do you take it every day? Do you only take it sometimes? What's the pattern?
So it's the default is you take it every day. It's a daily oral pill, and that makes sure that there's enough of the drug in your system that you're safe whatever happens. There is another regimen that men can take for Truvada, which is often referred to as 211. So that's actually what I do, where instead of doing it every day, you take 2 doses, so 2 pills, the day of sexual activity or some risk you're exposing yourself to. 1 pill the next day, 1 pill the day after, and then the rest of the time you just don't take anything. And that is easier for some people. And, that said, if I think about, if I do an informal poll in my head right now of my gay friends in San Francisco, I would say most probably do daily just because it's easy. I actually recently, my doctor tried to move me onto Descovy, which is the second one, just because it's better for kidneys. And when I was a baby, I had some kidney issues. That I do not believe doctors do recommend 211. And certainly my doctor didn't. And I asked him, well, I'm doing 211. Can I do that with Descovy? And he said, well, I'm not allowed to say yes to that. I said, oh, what do you mean? And he was like, I don't think that they've studied it with Descovy. And I was like, okay, so I'm just was sort of going to move on from that. Oh, and you're going to give me Descovy? And he was like, yes, I'm going to give you Descovy. So I'm in a sort of gray area on that. And anyone listening, don't treat me as a doctor and my recommendation on Descovy.
But so you've now moved to Discovery.
I'm in the process. But I mean,
Okay.
which is, I'm a fairly plugged in, good, health seeking behavior type person in the statistics. And I still haven't got around to being on the best one. So I don't know. It's if it's not top priority in a given month, I might not get around to changing to the better drug. I was talking to someone yesterday actually because I said, I'm about to record this podcast. What are you on? And he said, oh, I'm on the daily pills. Most people on the daily pills probably couldn't even tell you if they're on Truvada or Descovy. They both work really well. Doesn't matter that much. But he said, oh, I was thinking of going on to the injectable every 2 months. But then as I thought about it more, I have to travel for my job. So I was worried, well, am I definitely going to be back in San Francisco at the time I need to get the injection? Or is there going to be a 2 week delay where I'm somewhere else and then actually I'm more at risk? And actually, I think it's easier if I just do the daily pills. And I think that actually gets to how these drugs it can be complicated how they interact with someone's life and it's not just something you can read off a clinical trial of how useful they'll be. You have to think about, well, how is someone who needs this drug going to use it in their real life, and what there might be counterintuitive kind of pros and cons of having a big gap of 2 months between. That sounds great on paper, but there you go. Someone didn't want it because they wanted to just do it every day. And I actually think about this with treatment as well. If I have a friend who started dating someone who was HIV positive. He was HIV negative. He started dating someone HIV positive. And, now with the great drugs that we have, if you are positive and on treatment, you will be undetectable. You won't transmit the virus to partners, which is incredible. But my friend, he knew that, and he sort of rationally, but he was still anxious around sex because it was a scary topic, even if he could sort of rationally tell himself he shouldn't be scared. And then that can be a tough thing for a partner to deal with. And one day, his new boyfriend actually took his daily pills in front of my friend as a way to basically build trust. And that was a kind of beautiful thing to do. I think if I were in his position, I probably would have been offended and annoyed, and he was so generous. And then sure enough, my friend did build trust and fall in love and have, I think they had a wonderful sex life. But that is the kind of thing you wouldn't think about when designing a drug. It's like if you're on a treatment drug that was an injection that was, once every whatever period, well, okay, that particular trust building exercise would not be available to you. So, it gets so complicated. And then the people most affected by HIV and AIDS these days are, it affects women in Africa more than it affects men now, and especially in Southern Africa. And there the complexities are very different, and I won't be able to rightly summarize them. But for example, if, having to go to a clinic for a procedure or injection might be different than having to go to a pharmacy to pick up pills, which might be different than having pills on your shelf for many weeks versus having to go more regularly. And all of that matters a lot.
No, that's super interesting. I think one thing I was thinking about when you were talking about this was the kind of, what are the different problems that people will face apart from, okay, so we have this sort of struggle to, I don't know, schedule some of these appointments, things like that. I think one thing on that front was with some of these injectables, I think there's a kind of leeway that you have for when you get the next dose. You don't have to get it exactly, let's say 6 months or exactly 2 months after, but there is a little grace period that you can get it in. But that's still probably sometimes quite inconvenient for people if something happens, if they're in a different country or something. But there's the issue of, I don't know, taking a daily pill every day for many years. That might be hard for some people in terms of remembering, especially if it's a preventive pill. It's not something that is necessarily super salient to them if it's a condition they already have. But also it's this access, like what if the clinic is closed one day? What
Yep.
if something happens? What if, I don't know, someone takes your pills or they drop out of your bag or something like that? Like, how are you going to get the next dose?
Yep. And I can answer that for me, and I pick them up once every month. But I'm just one guy, and I think we need to get a better answer for people who are affected in other contexts. So I think it's time to phone a friend, Saloni.
We're going to phone a friend. This is so exciting.
We're gonna phone a
Who
friend.
are we phoning?
We're going to phone my friend, Douglas Chukwu, who works at Open Philanthropy with me. And before that, was a medical doctor in Nigeria and worked in public health on HIV treatment and prevention. So, should we dial him up?
Yes.
Hello, Douglas. How you
Hi,
doing? Thanks
Jacob.
for joining.
Good. Great to be here.
Well, thanks for taking time out of your day. So, we work together. But before we worked together at Open Philanthropy, you were trained as a doctor and worked on other things in public health, which is why we wanted to bring you on today. So yeah, what's your background and what were you working on before Open Philanthropy?
I trained as a medical doctor in Nigeria. So I had a couple of years of clinical practice working as a medical officer in a government establishment and also a private establishment. So had that dual experience and then pivoted to work in public health. Interestingly, most of my public health experience was in the field of HIV and AIDS.
Are people getting kind of a weekly stock of treatment or prep, or will it last them months or years? Maybe this varies depending on the type of drug that they're using.
Yeah, so oral prep comes in, like the commonest is in a pack of 30 tablets, and oral prep is to be administered daily. So the common, it varies from a range of 1 month to 3 months, 3 months being the maximum, because for individuals on prep, they need to be tested every 3 months as per the national guidelines. So that touch point with the facility, the 3-month duration of prescription helps to make sure that clients come for their refills, they are assessed for adherence, they're tested for HIV, and they are also monitored for side effects.
How important is public funding from donor countries like the US and the UK when it comes to HIV particularly?
Absolutely important, right? Let's say, for example, there are a lot of individuals that have accessed treatment that wouldn't have accessed treatment if the HIV programs in countries were entirely reliant on domestic funding. And this varies across African countries, but in Nigeria, for example, over 80% of the funding for HIV programs is via external funding, right? And, yeah, and then because there are some countries like South Africa where they've made some progress in terms of domestic financing for HIV programs, I think as high as 70%. But overwhelmingly in Africa, there are various country programs that are usually reliant on external funding for sustaining and delivering HIV.
We're recording this at the beginning of April, and I've still found it difficult to get good reporting on quite what's happening with PEPFAR, the US main way that the US supports HIV programming. And the answer of what's happening may change in the coming months. But as you talk to your friends who work in public health, what are you hearing at the moment? What has happened at facilities or on the ground in reaction to the PEPFAR uncertainty?
So the effects of this cut across not just the healthcare workers, but healthcare workers, patient communities. There's a lot of uncertainty. There's a lot of unease and a lot of worry about what the future holds. And a lot of these cessations were abrupt. So people got stop work orders. As I mentioned, there is a community component of healthcare service delivery. There are community healthcare workers that are supported by the PEPFAR funding, and having the stop work orders meant people stopped getting tested in communities. Access to some medications were threatened, even though perhaps there was some stock to sustain initial dispensing of ARVs. But clients were being told if this continues, you'd have to pay out of pocket for your medications, and that actually troubling for the patient community. Additionally, I think about the broader implication of this, which is knowing that the funding for HIV programs is actually threatened. That also affects manufacturers thinking about maybe wanting to exit some markets, and that kind of damages a lot of the progress that has been made over the past couple of years in the field of HIV and AIDS. There are, country governments rallying up to cover some of those gaps, but those resources pale in comparison to the amount of resources that the US government devotes to supporting this.
If someone needed treatments in the last few months or even now, how would the cuts and the stop works order affect them? What would they be experiencing?
I would say to paint a picture, imagine a status quo where every day community healthcare workers report to the facility, gear up with their mobile testing kits with their ARVs, and they go out into hard-to-reach areas. They identify people who are positive for HIV, place them on treatment. Some of these are pregnant mothers who don't have the resources to come to the facility, right? So that abrupt cessation means those individuals would not benefit from those services. Now, beyond those who are yet to be identified, because that's the category I just talked about, there are people who rely on these healthcare workers to reach them to receive their refills for ARVs, right? So the situation was abrupt, as you know. So people were just told to stop work, and there are clients who likely would be expecting their healthcare workers to deliver ARVs to them and would have been affected by such stop work orders. So that's pretty much, of course, there are those who still have drugs, but there are those who are actually suffering from these cuts.
In my I was reading a few articles about this, and the impression that I had was the clinics might have some of the treatment, but they're just shut, and there's no one. Well, the staff who are paid or supported by the U.S. are not allowed to go in, and
Absolutely.
people can't get treatment even if it's in the clinics there.
Absolutely. Yes. And the staffing, as I mentioned, the staffing support it's not limited to the community setting. Even in healthcare facilities, there are one-stop shops that are staffed by individuals that are supported by pepper funding. As you rightly point out, that stop work order affected those individuals, and perhaps clients who would have presented to facilities wouldn't have had maybe individuals to attend to them.
And I think we were talking about refills and how often people get refills, and if that's every 30 days or every 3 months or so, that probably adds up to quite a lot of people who have been directly affected by these cuts over the last almost, I guess, 2.5 months maybe.
Absolutely. Because people, it's a 3-month cycle. It can be a 6-month cycle for people who are stable, but every day marks someone's clinic appointment, right? So
It's a scary time for a lot of people with HIV.
How excited are you for Lenacapavir in the field? I think one reason I think it's going to be quite important is because of this adherence issue that you mentioned, but also meaning that people don't need to get refills as often, so there's a bit more stability for someone who has had an injectable. Is that also your view, or are there other things that you see as part of this?
one of the challenges with oral PrEP is having to take it every day. And with suboptimal adherence, there's the risk of resistance developing. So having a drug that's administered twice a year, I won't say it's as good as a vaccine, but there are challenges with the development of the HIV vaccine. So this is as good as we are currently towards making sure that people keep from getting infected with HIV. So it's very exciting in the HIV prevention landscape. Having an injectable once a year, fingers crossed, but that would be amazing. That would be phenomenal.
We're almost there.
That was really helpful. Thank
Thank you so much,
much.
Douglas.
Thanks so much. Happy to talk about this, and very excited about the developments in the HIV prevention space. Hopefully these developments continue and move the needle in terms of achieving epidemic control of HIV and AIDS.
This was so cool to phone a friend and learn about what things are like in treatment in Nigeria, the future Lenacapavir, but also all of the funding cuts and the disruption that's going on there right now. I think it really made me think of how important some of these new drugs could be in terms of changing around the epidemic in HIV. The possibility of using long-acting drugs, and by that we mean drugs that have an effect for a really long period. Currently, we have cabotegravir, which is a 2-monthly drug. There's also lenacapavir, which is a 6-monthly drug. And potentially, Gilead is also working on a once-yearly drug. And if that pans out, again, I think it would completely change our ability to respond. Whether that is actually scaled up is another question, and that's something we'll talk about later on. But it's really, I think it's a change in what's possible in treating and preventing HIV.
Yes, and I feel like I have a lot to digest, and we have a lot still to discuss. So let me go away for a second and think. Okay, Saloni, we're back. How you doing?
I'm doing great. It's been 5 days. I've got a lot to think about. You look like you're in a completely different place from before.
That's right. New shirt, new background. I am in New York City. I'm in the village, the East Village, or at least I thought I was. I arrived and was informed by someone who lives here that I'm actually in Stuytown, which is not the same thing as the East Village.
Oh,
But I've decided to kind of squint, and it feels about the same. And I'm having a good time. It feels. I wish I could say I was here, funded by our podcast to do some historical analysis of the AIDS epidemic, because I was in Casper before, and now I'm in the village, and those are two important parts to the story. However, I'm actually just here visiting a friend, totally unrelatedly.
I really enjoyed thinking about what Douglas told us about how treatment works in the field in a clinic in Nigeria, but also just thinking about the different approaches that people have to prevention, whether that's with condoms or behavioral changes or PrEP, this amazing breakthrough in 2012 of multiple drugs in a combination that can reduce the chances of infection. And it's really interesting first to think about the behavioral aspects that lead to basically how do people actually take these drugs in
Yeah.
practice? And how does that inform drug development? Like, how does that inform the kinds of new treatments that we need and whether they're effective? And I think that's ultimately the key breakthrough of this new drug that we're going to talk about, lenacapavir, that instead of being a daily pill that people would take as they do with PrEP, it's this long-acting injectable. So it's an injection that you would take. So there's, I guess, 2 injections every 6 months. And this massively reduces the chances of infection. It's also been used as a treatment for people with drug-resistant HIV, and there could be other purposes for it as well. So I think that's really a key breakthrough, and I think it really I really started to understand exactly how that would matter for someone in who has HIV, thinking about how do they get their next supply of the drug, how this makes a difference to them.
Yeah, it also got me thinking about the costs of how can we get the costs low for new drugs so that they can get used more. And that's something that I'm interested to talk about with lenacapavir too. But what is lenacapavir? Saloni, it's time for you to teach me something new.
So I think to teach you about what lenacapavir is, we have to kind of go back a little bit and talk about the capsid. So if you remember from before, the capsid is this thimble-like structure within the HIV virus that contains the RNA molecule, and it contains a bunch of other enzymes. And it's the core that stays intact when HIV enters the cell. And this capsid takes that information and those enzymes into the cell's nucleus. They then allow the RNA molecule, the genetic code of HIV, to turn into DNA, to integrate into our own cellular DNA, and then to proliferate into many more HIV particles. So it's this key structure that's kept intact throughout this process. Once it gets to the nucleus, it then starts to dissolve, letting the RNA molecule out, letting the enzymes out to do their jobs. And then later on, when the new HIV particles start to be formed, it then starts to form, the capsid starts to form again. And this process is actually really interesting and quite interesting both as a process, but also in terms of what it looks like. So this
Yes.
HIV capsid is made up of, I think, more than 1,500 or so proteins, but each of these proteins comes in groups. So some of the groups of this protein are in groups of 6, which are called hexamers, so hex is 6. And some of them are in pentamers. Pentamers, pent means 5. But there are 250 hexamers or so. It kind of varies. And exactly 12 pentamers.
I'm looking
And
now, and it looks like flower petals that are falling into place. How on earth is it so exact? Why are there 12? Why are there 12? And the 12 are not exactly, they're sort of dotted around in a pretty pattern, but not necessarily how I would have designed it if I were thermodynamic.
yeah, I mean, if you're listening, it's this thimble-shaped structure. Most of that is made of hexamers of this protein. So imagine some stick-shaped thing, maybe like a star-shaped cereal that I used to have
Okay.
called Honey Stars.
No, I never got those.
And oh yeah, and they
I was
were
stuck
very
on
tasty.
shreddies, which I'll never say a bad word against, but
And then in a few places in this capsid, there are other structures that are only 5, that only have 5 proteins. So they're like 5-star shapes. And these look, I mean, the where these are organized in the whole capsid doesn't look very symmetrical to me.
no.
And it's quite strange. But what I was reading was that the placement or the number of these pentamers within the protein within the whole structure changes the whole shape of it. It's sort of like the structure that's where the hexamers are tessellating. So they're all fitting together in this very symmetrical way between them. But then these 5-shaped pentamers determine the curve, I think, of the capsid.
Hmm.
Um, that's very interesting.
Yeah, I feel like if I redo the tiles in my bathroom or something, I want to do this. But now that you've told me the shape, the 3D structure is an important property that wouldn't be so good on tiles, so I'm gonna have to rethink.
You would have, maybe it would be more like a flower vase.
Yeah, oh, great idea, actually. Yeah, it's very floral.
Hmm.
It is very beautiful. I recommend people listening Google it. And I think recently, it's only the last 10 years or something where we've really known what these different monomers look like and how they assemble and all that. Is that right?
I think that's right. So only since the 2010s have we had advances in microscopy that allow us to see some of these particles with enough resolution to see what they actually would look like in this coherent structure.
Right.
And that is super interesting.
As I think about the drugs you've described previously, what is interesting about this one, if we're talking about the capsid and we're going to try and go after the capsid with a drug, is, you know, this is a structural part of the virus, but it's not as functional in the direct sense. I'm not imagining something getting integrated into my DNA, so I'm going after the integrase, and I'm not imagining, uh, you know, some chopping up the large string of proteins into smaller proteins and going after the protease. This is more like just a package, a thimble, a bullet, that it must be required because why would it still be there? But the function is not as direct. I don't know. Does that
Mhm.
ring true to
I
you?
think you're kind of right, but I think it is important because to make sure all of this stuff doesn't fall out, I guess, in some other part of the cell. Like we need to carry or
Yeah.
transport the RNA molecule and the other enzymes to the nucleus. But it also has these functions where based on the structure, based on the shape of this capsid, that allows it to enter the nuclear, the
Mm.
pores in the nucleus, the little holes, and this shape allows it to wiggle through. And I think it might also be involved in stimulating the reverse transcriptase step. So
Ah,
the
okay.
capsid is somehow involved in making that start. And I think that is also quite new discovery that people have had in the last 5 years.
Got it. Anyway, keep going. Thanks for pausing for me.
Not at all. Okay, so we've talked about what the capsid looked like. What does lenacapavir have to do with this?
Yeah.
This also, it really helps to have a visual, but I'm going to try to explain it um, in words as well. So we have this structure where the capsid is made out of these proteins. The proteins are sometimes in hexamers, sometimes in pentamers. I guess if you imagine that these hexamers or pentamers are fitting together, they're a bit like putting your hands together with your like knuckles, like they're kind of fitting together between your fingers. And imagine doing that lightly, like not, you're not fitting them too strongly. So there's a little bit of space and flexibility between your two hands. But then lenacapavir it wedges itself into those gaps
Right.
between both of your hands' knuckles. And that means that it now becomes very stiff. Like now you don't have this much ability to move the structure around. And this stiffness becomes a problem in several ways during the HIV's life cycle. And I think that's quite cool just to think about how the overall shape of this capsid changes based on lenacapavir fitting into these little hole gaps between the proteins.
That is cool. It's quite subtle as well, because it's not, I think of a drug coming in and trying to nuke some structure, blow it up. But actually, you're saying, no, we're just going to change the properties of how squidgy it is.
No, exactly. And yeah, I mean, I think it's fascinating. So we have all of these little lenacapavir molecules. Lenacapavir itself is quite a small molecule, so it fits into these little gaps.
Mm-hmm.
And if we go back to this life cycle of where the capsid is imported, so we have the HIV virus has entered the cell, has released some of its contents, which includes the capsid. The capsid is then trying to enter the nucleus. In order to get into the nucleus, it has to fit through these holes, the nuclear pores.
Right.
And to do that, it binds to certain proteins on the nuclear pore.
Mm-hmm.
And it turns out that those binding spots are blocked by lenacapavir.
Okay.
So it binds in exactly the same spots where those proteins would attach and let
Nice.
it squeeze through those gaps. And the second thing is the capsid is now too stiff because of lenacapavir
Right.
blocking. So it's less
My
flexible. It
knuckles are
becomes
engaged.
exactly. Your knuckles are engaged. You can't squeeze through the little holes of the nucleus. So that's another important thing. Then the next step, and this is super interesting. So it's not just that one step that lenacapavir disrupts, but it's actually multiple steps during this life cycle. So imagine that some of the capsids have still somehow made it through to the nucleus.
Mm-hmm.
Now the capsid needs to dissolve and allow the RNA molecule out and allow the reverse transcriptase to turn the RNA into DNA, and it needs the integrase to turn that DNA into a part of our own cell's DNA. But it now can't it can't dissolve, it can't uncoat anymore because of this rigidness.
Uh-huh.
It's just too rigid, but also sometimes it's so rigid that it cracks too early, and that early cracking is makes it hard for the capsid stimulating the reverse transcriptase to start doing that process.
Okay.
So that is super interesting. So we've now blocked it from entering the nucleus. If some gets through, it's now not able to release its contents or it breaks too early. And then there's a third part that lenacapavir disrupts as well. So imagine that you've somehow some of the virus particles or some of the capsids have still made it through, or maybe you're in a different part of the HIV virus's life cycle,
Mm-hmm.
and you're now trying to create these new virus particles,
Right.
the descendants of the initial one. And in order to do that, we have the immature HIV virus, and
I remember, yeah.
this now has protease involved, right? So we have protease cutting up these giant polyproteins into the proper form. And we have this new capsid trying to form to surround all of the RNA and the other enzymes. But what happens with lenacapavir is that because it's in this too stiff kind of formation, it's unable to form in the correct shape, and it just doesn't fully form.
Okay.
So there's this image here where you can see that the normal way that all of these proteins in the capsid form is that we have all of these hexamers and pentamers. They create these little clusters. They somehow self-assemble. Maybe this is just something that we don't understand yet, but somehow they self-assemble into
Which
bigger.
blows my mind just from if I'm visualizing the cytoplasm of a cell, I'm like, there's so much going on there. How do these all stay together and not get distracted? But anyway.
Exactly. So we're getting these clusters of multiple hexamers and pentamers. That happens, but now because of the stiffness, the shape isn't forming correctly,
Okay.
and the full shape just doesn't work anymore. So, I mean, I think this whole process to me was really interesting to learn about. I knew that lenacapavir somehow had this amazing effect at very low concentrations. It somehow has a really long effect lasting for at least 6 months. But I didn't really know about the mechanics of how that works. And the other thing I didn't realize until reading for this episode was multiple steps are involved. So it
Right.
disrupts this process at multiple steps. I think that now I would say this doesn't mean that it's impossible to develop resistance against it, but it does probably explain why it's so effective.
Hmm.
That it's targeting these multiple steps, it's reducing the probability that an infection can multiply. And so I think that was very interesting.
Yeah, I was gonna say it sounds almost like a combination drug itself. If you got three different stages it's acting at, but I don't know if the mutations that would generate resistance are correlated there or not. It makes
I
sense
mean,
to me though to have a fail-safe one, for the mopping up the capsids that try and form out the other end.
I think what I've learned is that there is still drug resistance can form, but it's something that forms if someone is on long-term treatment with lenacapavir. It's not something that they would have before the infection. And so it is still useful as a preventive
Got it.
sort of anti-infection tool. But as a treatment, there have been cases of people developing resistance, and that would be through exactly how this lenacapavir fits into the little gaps between
The gaps
the
start
capsid
changing.
proteins.
Yeah, I
If
mean
you
that.
change the shape of
Yeah,
that, yeah, that
it
might not
makes
just
sense
fit.
just from a selection pressure point of view. If you got many more viral particles, you're infected already, then you got higher probability probably of mutation.
Exactly.
That's good for HIV. Neat. I like it. How did it come about from a? Was this the first capsid inhibitor, lenacapavir?
This wasn't the first capsid inhibitor. I think people had been trying to target it for a while. And in 2010, Pfizer had developed another molecule called PF74. And that seemed to bind to this little gap, this pocket as well. And it could block and hyperstabilize the whole capsid shape. But it didn't work so much in the human body. So this drug, it was taken orally and it just didn't stick around in your body.
Hmm.
And so they gave up on the drug and they started working on other drugs instead. Instead, what happened was Gilead tried to build on this PF74. It seemed quite promising because you have a potential way of targeting the capsid protein, but we're just missing out on making it more available in the body and long lasting. So They did something called parallel synthesis. So they just tried creating lots of very similar compounds to that. So using the same molecule, but then adjusting it in lots of different ways. And the way that happens in the lab is you have lots of test tubes or plate wells where you have the initial molecule, the PF74, and then you run lots of different reactions in those different test tubes under the same conditions. So you have
Right.
maybe 3 or 4 with some reaction going on and so on. And they did a lot of iteration based on that. And it eventually resulted in Lenacapavir.
Got it. Well, thank you, PF74, for trying first. It's interesting. So it sounds like we had the idea and there was some binding going on and it was working okay. But then the practicality of the human body, you got a lot more steps. You don't just have to bind the part of the HIV virus you want. You also have to survive the machinations of the human organs.
And this is the really interesting thing that we'll talk about later in terms of how does this actually stick around for so long in the body? How does this have an effect for over 6 months? That itself is really impressive to me. I thought I could maybe just briefly talk about some of the different kind of iterations that people do.
I love that.
And how important.
To get to Lenacapavir, or
Yeah, exactly.
I'm in.
So we have PF74. Actually, don't you know someone who worked on this?
I do. I was wondering what to say, but I think I may have mentioned her earlier, and she and her PhD worked on PF74, not at in an academic setting, not at one of the companies, but I think knows a bunch of the iterations that you're about to tell me, but I don't know them because I should have asked her more questions first.
Yeah, I mean, it's so fascinating to know that there are people surrounding us that have been responsible for these huge breakthroughs, and they're just normal people who are sometimes friends.
I had to tell her I was about to talk about Lenacapavir before I realized that she had done so much of the work that led up to it. It's wild, yeah.
I think it's incredible. Okay, so we have PF74. What happens now? So we have all of these little reaction test tubes going on. One of them, they introduced a hydroxyl group. That's basically an oxygen and a hydrogen. And then they added an indole ring. That's a fused ring structure. So there's multiple atoms in a ring with a nitrogen group. So this massively increased the effect of the drug.
Okay.
But it didn't work in the body because it was broken down by enzymes in the liver.
Okay.
So, we've got this one. Didn't really work out. Then they tried another type of ring. This is a 6 structured ring with 1 nitrogen atom instead.
Okay.
This then improved how stable it was in the body. Like, it was not broken down very quickly anymore. But now the effectiveness was reduced.
Damn.
So,
I love how we're making atomic level differences here, and it's having. Anyway, keep going. Okay, so we tried that one. No luck.
they did a bunch of other changes. There were some, 7 or 8 compounds they made before getting to this breakthrough. And this actually comes from this really interesting book that I was reading called Drug Development Stories. And these researchers just put together how all of these different types of breakthrough drugs in the last few years were developed. And so this last
there are a few books like that I'm trying to. I can't remember if I own that one, but they're fun to flick through. So I'm gonna have to check my bookshelf when I get back to San Francisco. If not, it's going on the order list.
I mean, it's often hard to find the exact stories about behind how these drugs are developed, and I assume, part of that is because it might be some kind of trade secret or something like that. But when I do come across someone writing a retrospective, or someone giving me the details, it's just a completely different picture. Like, it really helps you understand exactly what they struggled with, how they were thinking about the process, and so on.
So if you're listening and you're working on drug development of some form, and you're wondering, oh, if I write up my process, is anyone even going to read it? We're going to read it. Write it up. Saloni needs it.
I need it. I love it. Okay, so now we have this breakthrough compound, I think, which might have been the ninth one that they made as an adjustment. They now replaced the amide group with an amino-indazole group, and that is a ring.
Amino-indazole.
It's a ring with 2 nitrogen atoms. And this improved both the potency, how well it fit, and also reduce the level of breakdown in the body. This felt like, okay, we're making something that that effectively tracks both of these key things that are important in drug development.
Got it.
Then it was just slight adjustments, slight tweakings from there. They added another amino group. They changed the placement of the amino group. They introduced a sulfone group that's a sulfur with 2 oxygens. And that was what resulted in this very highly potent molecule that was very stable in the body. And that became lenacapavir.
We made it. Highly potent, highly stable. And, it's funny. I think when you say the body's not breaking it down well, to me, it sounds like, oh God, the body's not breaking it down well. And then in fact, we've been seeking a molecule the body doesn't break down well because it can last for longer and protect you for longer.
I mean, it's different purposes, right? So, I mean, if you're trying to reduce maybe the side effects of some drug, you want it to
Right.
get broken down very quickly, just have its action disappear. But if you're trying to develop some drug that has a very long-lasting effect, then you want it to stick around for a long time. And so, I mean, finding something that is both very safe, very highly effective, potent, and also very stable in the body, that makes a great long-lasting drug.
That's the trio. And we got there.
The other interesting thing I learned about lenacapavir, and I think you might maybe come to this later on, but it's the drug with the most fluorine atoms in it that's approved by the FDA in the US. So fluorines are often used to increase the stability, I think, in the body. And lenacapavir has 10 of these atoms in the whole molecule. And usually that has led to drugs that are unsafe in some way. But in this case, it has very high safety profile as well.
I remember seeing that, and I'm not enough of a chemist to tell you why there aren't more fluorines. But I do like the idea that there's fluorine in the aqueous solution. There's fluorine in the water,
What did we talked to your friends Sanela, is that right?
Yeah.
Was there stuff that you learned from her on lenacapavir and capsid inhibitors as well, beyond what we've talked about?
The thing that she emphasized to me was really in line with what you just said about how iterative the process was. That starting with PF74 has a great binder. There was a lot of tries of just getting the other properties that could make this into a useful drug. And so I think the thing she was really struck by was the PK, as drug developers say, looking really good for lenacapavir, and
What's the PK?
the pharmacokinetics, I want to say. And there's, again, this will betray that I'm not a medicinal chemist, but pharmacodynetics and kinetics and pharmacodynamics are the 2 things that you'll hear people talk about all the time about how the body processes drugs. But I mean, also she was really emphasizing it that previous molecules' stability was an issue. And then, seeing lenacapavir being so stable and so low toxicity, such a small amount, and it can stick around for 6 months. It's, getting to the 20th or 15th iteration off of the initial principle, and then getting those great properties was, it was wonderful for her to see as someone in the field. And as someone outside of the field, I didn't realize I'd heard of lenacapavir as this miracle drug, and I didn't realize, of course, what came before. And there's a lot of steps to get there. It's not that you suddenly come out swinging and suddenly discover the exact perfect thing.
Mhm. Right. I mean, the thing that I remember, I did my undergraduate degree in biomedical science like 10 years ago, I think. And I remember a little bit about pharmacology, just PK, as it's more you're measuring both how fast is the drug broken down,
Hmm.
how much of the drug is broken down, how quickly, but also how available is it? Like
Yeah.
how much does it actually get to the organs that you need it to get to? Is it able to have its effect there? And I think with lenacapavir, it seems to be very effective even at small doses. So if you imagine that over time, so you have this injection first of lenacapavir, and then over time there's this decaying exponential curve. So it just quickly starts to break down, and then that breakdown process slows down. But even at the very low levels, it's still very effective
Yeah.
and able to block this little site within the capsid proteins, and that is what makes it so effective.
Well, let's do a quick detour, if you'll let me, on long-acting drugs other than lenacapavir, because the principle we're talking about applies not just to this molecule. So the nature of a long-acting injectable or a long-acting drug, well, what makes it long-lasting, what makes it long-acting? It's relative to what we're used to, relative to immediate release drugs. If you think about the PrEP drugs that we talked about earlier in the episode, those you take daily, and that's because you want to have enough of the active ingredient in circulation in case HIV enters your system during that period. But pretty quickly, most drugs get metabolized and get filtered out and leave your body in urine and sometimes other ways, but that's the main one. And so an issue that you mentioned is if you have to take a drug every day, you might forget to take it. You might run out of the drug and not have time to get a refill for a few days or a week. Or I actually behind on refills because I'm in New York right now. And or you might not want people who you live with to see that you take pills regularly. So keeping them around is uh, not not ideal.
Mhm. This
And
is important with HIV as
yes,
well
absolutely.
because some of the preventive drugs might also look like treatment drugs. I think you mentioned this earlier. And
Yeah.
so there's also the stigma around people thinking that you have an HIV infection and then they're worried about that, even though these drugs are very effective and it's very unlikely to transmit if you're using these drugs. But I think this whole thing of how do people actually take it in their daily life
Yeah.
is so relevant for drug development.
Absolutely. And the long lasting prevention that people might be most familiar with is birth control, where a lot of, some similar issues are a big part of what can drive different women to want to make different choices with birth control. So, the pill was first approved by the FDA in 1960 after development in the 50s as daily oral contraception. And then from the 60s onwards, there was a lot of work to see if you could make the different options for birth control long lasting. So, can you have hormones that are inside a silicon tube of some form that you can control the diffusion over time so that you
Okay.
don't have to take daily pills or take daily hormones? And now there are multiple options for that. So you can have hormonal IUDs that release progestin slowly over time. You can have arm implants. The the approach there is to take an existing biological molecule or synthesize an alternate of a hormone and then have a different packet that controls the diffusion. And so it's a bit different than what we just talked about with lenacapavir, where the drug itself is so insoluble and unstable, and that it itself sticks around. Whereas if, let's say, you have a drug that doesn't naturally stick around, well, maybe you can design a delivery mode, a polymer, a liposome that you can keep it inside of and slowly diffuse, and you can achieve the same goal. Maybe you could just put it in a drop of oil, or you could suspend it in some other way. Maybe you could put it in a device. And sure enough, for HIV, one new mode of prevention that we haven't talked much about yet is our vaginal rings, where you can have a slow release. You can insert a vaginal ring monthly, say, and have a slow release that a woman who wants to not be at risk of HIV has more control over than if you have to get, in some circumstances, than if she had to get pills that were more visible to people in her household or her husband or something like that. So there are a lot of different strategies here. When it comes to HIV, what's interesting to me is that there are a few long-lasting drugs that are being tested now, or are near the finish line now, that use different approaches and yet achieve this kind of similar goal. So the three that come to mind for me are lenacapavir, which we've been talking about a lot, but then also cabotegravir, which is already approved in some countries, in the US, which is this injection every 2 months. And then also islatravir and other follower drugs that are being made by Merck that are oral drugs that you might be able to take once a month for prevention or for treatment. Maybe you take once a week. There's a few different regimens being tested. And my understanding of why islatravir sticks around for a month is...
Which one is islatravir?
Islatravir is a drug...
Is that Merck's?
Yes, made by Merck. And, imagine instead of taking daily oral PrEP, you take one pill once a month, say, and they've tested that regimen. And what happens after you ingest that pill is that the active ingredient sticks around intracellularly. So, it's not, when you're imagining lenacapavir, what you should imagine is you've got a drug substance dissolved in a liquid that's 40% water, and you're getting injected with that. The liquid's kind of dispersing, and the drug is sticking around and forming a solid. And I, on the video, just point at my arm, but it's not in your arm, it's your stomach or in your butt. And then that active ingredient is slowly dissolving over the course of many months. Whereas with islatravir, you should imagine that the active ingredient is in some way, and I wish I knew more about this, I don't, is in some way sticking around in your cell, so that if the HIV virus is entering your cell, it's gonna do its work, which is wonderful. But it's not, some big depot, some big lump. It's totally different, and it might stick around for different biological reasons.
That is so interesting. I had no idea. I was going to ask about, this container or the package. Does the package dissolve? Is the package is that actually a separate molecule or something like that? But you answered my question. I guess that also varies with other drugs.
Yes, it does vary with other drugs, and the answer will differ for other drugs because there are different ways to achieve the goal. And if you don't get a drug like lenacapavir that has properties of slow degradation, then you might want to achieve the goal via packaging your active ingredient differently. One thing that I think's in a way fortunate about both lenacapavir and islatravir and cabotegravir, which is also a injection that's a sort of suspended solid that slowly degrades, is that you don't need a very complicated package, because a complicated package adds complexity, manufacturing cost, and makes me less optimistic that in the near term a drug will get used in lower resource settings. Whereas the simpler you can make the package, the more likely you can use a drug in many settings. And in this case, it's just the drug itself. Well, not I'm simplifying a little, but it's mostly the drug itself in a syrup solution. And you know what? I could take a look at that. I might be able to one day inject myself with that. So
This is so interesting. I mean, the other thing that reminded me was, I think what I read about the fluorine atoms is that those help it stick around in your fat. So you have the injection either in your abdomen or your butt. And then I think the fluorine atoms keep it around the fatty areas. But also they create this like this little lump
yes,
underneath your skin, right?
yes. Well, I mean, I think I absolutely, and as I think about the lump, I was just thinking about it spatially, and I would love someone who's worked on injectables and on Lenacapavir to correct me in the comments if I'm thinking about it spatially wrong. But, is Lenacapavir a 1 or 2 milliliter injection? Do you happen to recall?
Yes, it is 1 to 2.
Okay, so my memory of how milliliters work is that's 1 cubic centimeter. Is that right? Okay, so now I'm visualizing. We've got some liquid that's 1 by 1 by 1 centimeter. That's quite a lot. And, you get injected somewhere and the liquid does sort of disperse. So you've only got some of it left. But a good amount of what you're getting injected with is this high concentration drug that becomes solid. And so sure enough, you should expect that to be a lump. You just got injected with a decent volume of stuff, and fair enough, some of it precipitates and becomes solid. And if there wasn't a lump, I'd almost be confused. I'd be like, where did it go? Yeah, I thought it was meant to stick around for 6 months. Anyway, so I think lumps form differently for different people. But
I think what I read was also that the first injection typically leads to this lump forming, but the subsequent doses don't. And so that makes me think that maybe there's some bodily reaction to the drug substance that creates the lump, and that the second or third time your body gets used to it in some way, or it just doesn't have the same reaction. But that was also really interesting because it reminds me a little bit about when people had smallpox vaccines, you could see that from the little mark on their shoulder or their arm. And with this, you have this tangible little bump in your butt or your stomach that typically shows you if the drug is from this little depot.
Yeah.
I'm curious about these other long-acting injectable drugs that you've read about. So, how else do they differ from regular drugs that only last a short amount of time? Are they more expensive? What's their safety like? What's all of that?
Great questions, and they again are going to have variable answers rather than an easy one. It depends on what package you're including. On the safety front, you really want to have, if anything, higher safety standards when you're doing something long lasting, because with a drug that washes out of your system within a day, that there's only so bad it can be. But with something that sticks around for a long time, you want to make sure it's harder to get rid of. So you want to make sure that before you get injected or before you take it, there's not going to be toxicity or longer issues. I think that is part of why with Lenacapavir and with other drugs, you get an oral lead-in for 2 days before you get long injections, just to check tolerability. And it's interesting, though, on efficacy as well, that you can sometimes have the same underlying chemical that is trying to achieve some, goal, medical goal, but it can be higher efficacy in a long-lasting injectable for a couple of reasons. One that we've mentioned is the real world and having, it's much more reliable if you just have to get one injection or swallow one pill than if you have to remember to do it all the time. But another is more chemical or more to do with the body, which is that when you take daily pills, you get this spike in how much of the drug you have relatively soon after you take it, operating in the bloodstream or elsewhere, that then decays relatively quickly. So you're kind of doing this spike and drop, spike and drop, spike and drop for daily drug, and that's not ideal. Often you want to be in a therapeutic window that is not so spiky. And with these long-lasting injectables, you have a lot more control and you can tune that a lot more easily because you can predict, okay, on a given day, how much am I going to have in my bloodstream based on how much I was initially injected with and how much time it's been since then. And you can really hit that therapeutic window perfectly. So you can end up with a chemically more effective drug. And, you as I said, you want to check the safety a bit more there.
That totally makes sense. So the therapeutic window, that's the range of the, I guess, the volume or something of that drug in your system, and having that in the ideal range, right?
Yeah, you want
Um, and I guess the other thing I was thinking about was maybe it's not just about the predictability, it's also that there's less of a fluctuation. Maybe some people react badly to really, these spikes, or the lack of, if the dosage suddenly drops, does that mean the effectiveness is now not high enough? But if you can manage to get a stable level of this drug for a really long time, that is probably better in some respects.
Yep, absolutely. And my mind goes to what other diseases for prevention or for treatment other than HIV would you want those properties most? And that's beyond the bounds of this short podcast. But the ones that I can't wait to learn more about myself. So malaria is one where if you're under 5 and live in West Africa, so if you're a kid in West Africa, you will get preventive malaria drugs during the rainy season where you're most likely to get malaria. That's about 4 months long. And currently, kids will get, 3 days in a row worth of drugs each month. So that's sort of 4 times 3. And the drugs they don't taste great, or, kids don't always like them. And, don't always take them all 3 days in a row. And, if you're a busy parent and your kid is making a scene, then you might not make sure to force all 12 of those doses. But if you could get a long-lasting injectable for a season that could cover that season and make sure you had the right amount of preventive drug in your system if you got bit by a mosquito, my goodness, that could be an enormous deal. And I don't think, based on my knowledge of malaria drugs, that we're close to rolling out something like that. But I do know that people are working on that problem, and it's very interesting. I think that tuberculosis is another area, hepatitis C is another area, rheumatic heart disease is another area. So a few infectious disease areas that I'm sort of, I'm getting excited about, but I don't know in super depth. And then beyond infectious disease too, there's a bunch of potential applications.
I think there's a psychosis drug. So there's a schizophrenia or bipolar disorder drug, right, I think, that's also long acting. What I was thinking about was maybe it's useful for things that are hard to predict when you're going to be infected by them. So you would prefer to take something that lasts a long time.
Yeah.
And so for various infectious diseases, that seems useful. Maybe also for chronic diseases where you have the condition for a really long time.
Yep.
Therefore, it's useful to have this long-acting treatment instead of doing something where you have to take it every day. But I mean, that kind of covers most diseases.
There's a lot
seem
of
like they
diseases
could be.
that description. And that, you should also think about, when do you want something to end? And I think for a lot of those diseases, you would not want a drug necessarily that lasted for life if you could have one stick around.
That is very true.
So for some drugs, for example, if you think, oh, there's a chance I want to get pregnant in the next few years, then you might want more control over when the drug's out of your system in case that drug hasn't been tested as much in pregnancy, or has been tested and isn't as safe in pregnancy. There's, one thing that in the literature that I've found interesting to read is the limitations in children of long-lasting drugs. And I was wondering, oh, that's such a shame because of, for example, the malaria thing I was just talking about. Why is there a limitation? And one reason is that the dose that you get given of different drugs is relative to how big you are. And the correct dose
Right.
is often bigger, the bigger you are. The trouble is with a child that in 6 months, their size is going to change a lot. And that means you might want to be dealing with a different drug dose in 6 months' time. So there's reasons like that you don't want to last forever. And that contrasts usually with vaccines, where you actually wouldn't mind having as long a lasting memory responses as you can. And there are some vaccines that are known to be pretty poor at generating long-lasting memory. So flu vaccines are sort of a famous example. The malaria vaccines so far aren't that durable, but are getting better. And then the best vaccine immunologists talk about is yellow fever vaccine, where you could live for 10,000 years and you wouldn't be getting yellow fever after you got that vaccine. And
Wow, I didn't know that.
yes, well, that will have
I
to.
can live for 10,000 years.
That's what I've heard, but I don't know how they extrapolated that. I think you can live for 10,000 years, and I think you.
I mean, I kind of want to live for 10,000 years now just to see this vaccine lasting that long.
Imagine if you got a challenge with yellow fever at the end of 10,000 years to prove it, and then it actually got you, throwing up black bile and everything else.
The other thing I was thinking about when you mentioned that on the size of the body was with lenacapavir. So what I understood was, okay, the fluorine atoms keep lenacapavir in fat tissue. But again, this is a problem for the same reason I mentioned. If your body size changes, that could affect how quickly it dissolves, I think, or how quickly or how much is remaining in this little teapot. And so imagine if you lost weight for whatever reason, you're on a diet or something, and
Yeah.
somehow, suddenly, this drug becomes more potent or effective in your body. I just thought that was kind of a funny thing that I hadn't thought about.
No, totally. I mean, another one
but
is it's hard to predict ahead of time is, are you going to want to go on another drug for another reason that might have a negative interaction with a long-lasting drug you're already on? And my understanding of lenacapavir is they haven't discovered many drug-drug interactions that are that concerning, but that might apply to other long-lasting drugs. So you got to think about that.
That's a really good point. The thing that reminds me of is grapefruit juice. Have you read
Really?
about this thing with so grapefruit juice has this chemical that interferes with the liver's metabolism of lots of different drugs. And
I had
if you
grapefruit
were
juice 3 days ago. I'm nervous.
no, did you?
Yeah. So that
Well,
explains
it's
why
not. I
I'm
mean, it's
so.
not all of the drugs, but it seems like it's quite a number of drugs that are affected by this. And so, if you're drinking grapefruit juice for whatever reason, it sometimes makes various drugs last longer in your body because it
Oh, wow.
interferes with their breakdown. This is true for I think some other chemicals and drinks as well. But that's the most commonly known in medicine.
So if you're getting towards the tail end of your 6 months of lenacapavir, just start doing shots of grapefruit juice.
I don't know if it interferes with lenacapavir specifically, but it seems to be a bunch of other drugs. But I mean, it's just an interesting thing to think about because now you're on this 6 monthly drug. What are the things that you now have to be thinking about to make sure that this drug is still working as expected? It's great to hear that it doesn't have many drug interactions, but I think that's the other thing that I like. If I was a drug developer, I would be thinking about.
And I'm sure there's more still to learn about drug interactions, so we'll find out. And, the reversibility of some other preventive tools is a key part of why people want to use them. So for IUDs, for example, you can get an IUD removed and it will not affect your long-term fertility. Whereas for lenacapavir, you got to wait it out. Once it's in there, it's in there.
I would find that exciting, but yeah, you're right. If there's something that could go wrong, that is something that this is why we need these, long clinical trials to make sure that this drug is safe in the way that people take them in their daily lives. Maybe we should talk about the clinical trials and how.
Yes, I was wondering about the clinical trials. How did we confirm that lenacapavir does actually work in the way we hoped, and that it is safe? And where did we go once the scientists had done the experiments in the lab?
I think the period when lenacapavir was developed was the late 2010s.
Yep.
They did some molecular studies, developed lenacapavir. They then did a phase 1 study, which is you're testing the safety in a small number of participants, and you're checking basic things about the properties, the pharmacokinetics, how long does it stay in the body, how effective is it in very specific ways. With a small number of people, I think that's usually like dozens or so. Then after that, they moved on to phase 2 trials. This is a second part where it's a larger number of people. Now you're testing a little more about the safety, because now you have like a wider range of people with different backgrounds. They might be taking other drugs at the same time. They have different behaviors and so on. So you can find out a little bit more about the safety. But also now with a larger sample, you can see how effective the drug is. So they did this with people who already had HIV and were taking other antiretroviral drugs.
Okay.
And they saw how this combination of lenacapavir plus those drugs works. And then the breakthrough that really got me to notice this drug was their phase 3 trial. And their phase 3 trial was, I think it started in 2021.
Okay.
And they had 2 different trials. One was with, I think one was with men. The other one was with cisgender women, specifically adolescent girls and young women. So this is quite important because the transmission and the effect of these drugs can vary for women who are trans, because the root of infection are sexual activity, but how that actually works is different. So they're focusing specifically on cisgender women aged between 16 and 25.
Okay.
Girls who are not using PrEP, they hadn't done HIV testing, or they hadn't done it in the last 3 months. And I think it was several thousand. So it was around 8,000 women in this trial.
Wow.
Across Uganda and South Africa. And this is important because if you're trying to test how effective a drug is, you need there need to be enough people, at least on the placebo, who are getting infected, so that you can see what the difference would be with lenacapavir.
Yeah.
So they focused on these areas where HIV incidence rates were relatively high, meaning that more than 3 or 4 people per 100 people were being infected per year. So imagine 3% of the population of this age is being infected with HIV. That's really, to me, that's really high. This is quite common in some areas of South Africa and Uganda. And that means that it's much easier to tell if lenacapavir has an effect.
Because you will detect in the other arm of the trial that there
I
were
mean,
HIV infections occurring.
right. If you imagine, okay, if you were doing a trial in the UK or in the US where people are already taking PrEP, or the rates of HIV is just so low to begin with,
Yeah.
then imagine no one in the placebo group gets HIV. How are you going to tell if lenacapavir is better than that? There's nothing
You
lower
won't
than
be able
zero.
to distinguish because happily its transmission is lower than it was 20 years ago or 30
Right.
years ago. Yeah, it's interesting. I mean, it's a very clear case, I guess, of trials in, well, I'm in the US, you're in the UK, but I may benefit from this drug living in the US based on trials that occurred in other countries because those trials were able to, they were in higher transmission settings, so you can get a statistical answer to the question that would have been harder if you were just doing the trials in the US. So that's a kind of selfish benefit that the US and people like me get from the global nature of clinical trials.
And that's what I think makes it really important to, we'll come to talk about this later on, but this is what makes it really important to think about how to actually get it to be accessible, how to scale up this drug in the future to the people who need it. This, wasn't developed without the help of all of these participants who agreed to be in this trial,
Yeah.
who are living, thousands of miles away from us, and who are responsible for this breakthrough being tested and the fact that we know that it works and so on. One of the other things that I found quite interesting about this trial is how trials actually work in terms of the healthcare and the screening involved. So, I mean, I think it's when people think of a trial, maybe they don't realize that you're not just receiving the treatment itself, but people are doing other types of screening to, monitor how you're responding to the
Yeah.
drug. But they're also, in this case, doing tests and screening for other types of related diseases. So what they did in this trial was they provided individual counseling to people. They provided condoms, lubricants. They had support for reproductive health in general. They also provided treatments for other sexually transmitted infections, and they did routine tests for some common sexually transmitted infections like chlamydia, gonorrhea, and syphilis.
Nice.
Which means that by participating in this trial, not only do you get the potential of this drug and also the, potential side effects or risks of participating in trial, but you also get the actual health care
Right.
because of the clinical trial setting. And I think that's something that people might not realize, that we have this system where, if you have these clinics or these hospitals that are running these clinical trials, they're also providing care to the people in the trial. When we think about how to actually set up these clinical trials in countries in Africa, it's not just about doing the testing. The researchers sometimes are involved in setting up new clinics. They're involved in
Right.
recruiting staff to
Yeah.
work on all of this testing and stuff like that. And that has various other benefits for the people in the area.
One thing that we've worked on at Open Philanthropy is an area that's not HIV but relates is congenital syphilis, where if it's syphilis is nowhere is it incredibly prevalent, but in some places maybe 1 to 3% of people have syphilis, and that is not something you want for yourself, but it also is really bad if you're pregnant because you may have a birth complication basically, or a miscarriage, or being born with congenital syphilis is very dangerous. And one thing that HIV care has brought in many countries is better antenatal screening, prenatal screening for HIV for many women. So you'll get at least one visit with a doctor, whereas in many areas before, if you were pregnant, you might not have even had one visit with a doctor. And so that infrastructure set up to screen for or to give you a touch point and some care while you're pregnant, mostly funded by the HIV world and by PEPFAR and other donor countries, has enabled screening for syphilis as well. There's now a dual test where it's a rapid test. It costs just under $1. You can screen for HIV and syphilis at the same time. And then if you are positive for syphilis, you can get relatively easy treatment on penicillin. And that's such a clear example of the benefits of this infrastructure that, were not initially planned from when HIV donors made those investments, but that spill over. So it makes sense to me that not only are there benefits from the knowledge gained of these lenacapavir trials, but people in the trials got better care too, and there's now probably better trained doctors in the area and that kind of thing. Yeah.
We have thousands of young women in this trial. First, they were tested for whether they already had HIV at that point. And this was useful, as I'll come back to later. This is useful to know what is the rates of HIV in the population. And it turned out that it was about 2.5 per 100 people per year, which is so 2.5% of people are in this age group are infected by HIV per year, which is, to me, really difficult to think about. Those women would not necessarily benefit from this treatment. That was not the purpose of this trial. It was to find out whether we could prevent new infections. So those participants were not included in the rest of the study, I think.
So you screen at the beginning for HIV. If you're already positive, then this is the trial for you.
Right. And the women who were HIV negative were then randomly assigned to getting either the Lenacapavir injection or I think this is Descovy. It's the oral PrEP pill, which is emtricitabine and tenofovir alafenamide.
Right.
Or the third option was FTDF. I think there's another name for this. Is that Truvada?
That's Truvada, yeah.
That's Truvada. That's also an oral pill that that people take daily. That is also emtricitabine, but this time it's tenofovir disoproxil fumarate. And I think what they did was, because one of them is an injection, the other two are oral pills, they actually gave like fake version of the opposites to all the participants so that they don't know which one they're getting.
Okay.
So they're all getting an oral pill and they're also all getting an injection. But some of the people who are getting the injection are getting lenacapavir. The others are just getting a placebo, which is just some water or something like that.
Which, I'm already curious about 3 things from what you said. Number one is if I get the fake lenacapavir injection, does it form a depot, and can I tell that it's actually fake because it doesn't form a lump? But my other 2 reactions are maybe more fundamental. So it sounds like there's no placebo here in the sense of there's no one who's getting no drugs, because that would be unethical. We already have drugs that we know will reduce your chance of acquiring HIV if you're on them. So the arms that you described are the lenacapavir arm, Descovy arm, Truvada arm, and there's no 0 arm.
Right. And I mean, in other clinical trials, you're not necessarily preventing people from getting other treatments. They could be taking other treatments for the same disease at the same time. But in this case, they wanted to see how effective was this as a prevention. And to get enough statistical power, you need everyone to be in the trial. And you're having this situation where you don't want any of them to be taking PrEP that you can't analyze in a consistent way.
Okay.
So it's both ethical in the sense that they're providing all of the participants with one of the 3 PrEP drugs. But it also helps because it makes these comparisons simpler. So they're not taking an additional PrEP, like some of them are not taking an additional PrEP drug that could complicate the analysis.
Okay, got it. Makes sense. And then my third reaction was, it's interesting that Descovy was in the mix, because I thought that Descovy was not approved for use in cisgender women.
Yes, you're right. It hadn't been tested before in women, and they had,
Ah.
and the company Gilead had been criticized for this having this approved but not testing it beforehand. So this also functioned as not just a trial for lenacapavir, but it also tests how effective Truvada and Descovy are
Great.
in the same
Okay.
population. So you get to have the answer to 3 questions. You have the answer to how effective lenacapavir is, Descovy, and Truvada.
And then because of what you said about the screening on the way in, can they compare it to what they think the background rate, if you're on nothing, probably would be?
Yes. Yes. So
Okay.
actually, you have four things you can find out.
Well, that one's not measured. I guess that one's kind of
Right.
interpolated.
So you can now compare these 3 drugs, but you can also compare them all to not taking any drugs.
Okay, well, I feel I'm on the edge of my seat. So, what were the results?
What were the results? I'm going to show this chart. So this chart compares the outcomes in each of these groups. So the first bar is showing the background incidence. These are the women who tested positive at the start of the trial, and around 2.4% of them got infected with HIV per year. In FTAF, which is Descovy, that had a rate of 2.02.
That's
And
very
in
similar to the background rate.
Truvada, it's around 1.69. Like these numbers are the point estimates, and that's our best guess. But there's uncertainty around just what the number is.
Sure.
So they all roughly fit into the same range. So
Yeah.
without drugs, and Descovy and Truvada have similar rates of HIV infection. And I think the reason for this is because it's hard to take these on a consistent basis over time. These are daily pills where the problem is one, maybe remembering to take it every day. Second, having enough supplies with you every day. The stigma that we talked about, maybe these issues around getting a refill on time. So there are all of these issues that make it difficult to take these drugs in the long term for women in Uganda and South Africa, where this trial was done. And what was interesting about the study is that they could actually measure how regularly people were taking these drugs through blood testing.
Because they're taking samples. Wow.
They're taking dried blood spot samples
Okay.
from people, and then they're testing the level of tenofovir in their red blood cells. And so this directly tells them what is the concentration of this drug in this participant. And with this, you can see how over time, the expected, how frequently people are taking them, that reduces over months of the study. People
really?
on average are taking them quite often, but over time that adherence gets much lower, so they're mostly taking them 1 or 2 times per week by the end of the trial.
I see.
And this gets back to this previous chart. And so we've seen what happens with Descovy and Truvada. What happens with lenacapavir? 0 women out of 2,134
My God.
get infected with HIV.
Oh.
That is just an incredible result, and there is some uncertainty around that. So the efficacy, so how much lower the rates of HIV infection is compared to the background rates, is 96 to 100%. So it's somewhere, it's not completely effective necessarily because there isn't a large enough sample to say
Right.
that this is a
We can't rule out.
100% efficacy
Right.
rate. But it is more than 96%
That is
of a reduction.
so incredible. That is so incredible. 0 cases.
0 cases. I was reading about this, I think on Stat News, the health and medicine magazine, and they mentioned how these results were presented at this conference, and they just got this standing ovation where people, I mean, unsurprisingly, this is just an incredible result.
Unbelievable.
One last thing about lenacapavir, Truvada, and Descovy is the side effects. So we talked about, okay, we have this long-lasting drug. That means there's a risk of long-lasting side effects as well.
Yep.
What happened in this trial was they didn't find that much of a difference between the different groups. So most of the side effects were similarly seen in the different groups. And those are mostly things like headaches, fevers. But again, when we record side effects in a trial, we can't necessarily conclusively say that these are because of drugs. People have headaches
Yep.
and fevers,
And we don't have a clean placebo
typical.
here, I guess. I mean, it's
Right.
interesting that when I think about what friends report as side effects of oral PrEP, because it's oral, there's often digestive issues or stomach problems. I wonder if they tested for that, because I would guess intuitively that lenacapavir would have fewer of those ones, but I don't know if they tested.
And you would be right. They
Oh, okay.
they did find lower rates of nausea and vomiting
Oh, there you go.
with lenacapavir. And I guess this is because of the difference where it's not an oral drug, it's an injectable. So it's not passing through your digestive tract in your stomach.
We don't need to do trials. You can just quiz me on my guesses.
The other thing was the little bumps on people's skin,
Yeah.
the depots of lenacapavir. So that was quite common. So about 70% of the people who got lenacapavir developed these little
Okay,
bumps,
yeah.
nodules below their skin.
Yeah.
And those typically, shrunk down to normal after a while, but also the next doses tended to not, you wouldn't tend to see those.
Yeah, I'd love to learn more about that, because to me sounds like success. I want a little nodule. I want that. I want to know that drug is there, and sure enough, in 6 months, I want the nodule to be gone because the drug is gone. But I guess there's more going on in the body than I'm projecting.
And I was also surprised that 30% of people who get lenacapavir don't develop these nodules. Like what is causing this difference? And I sadly don't know the answer to that, but it's quite interesting.
So and I was about to ask about other trials outside of women, but is there anything else on this trial that I should know first?
I think maybe we should talk a little bit about, why was there almost no difference between Descovy and Truvada and the background rates? And why is it that lenacapavir is so effective in trials or like in the real world? I think I would say that it's not entirely clear how effective Descovy and Truvada are compared to not taking any drugs. And that's just because the uncertainty on those is fairly moderate. So there isn't a very precise figure that we would have. It seems like they're roughly similar, but there could still be some meaningful reduction that these are providing. I guess the other reason is if people are not taking it regularly, these oral pills regularly for whatever reason, in the long term, that reduces the efficacy. So even if someone was taking it every day, it would be a higher level of effectiveness than in the real world where people are taking it less often.
Right.
And I think this is why lenacapavir is so much more effective. It's not just that it's highly effective on its own, but it's also really long lasting. And that both of those contrast with Descovy and Truvada.
Yeah, I guess it's proving the hypothesis with data. That's what we were wondering, and we were hopeful that lenacapavir's long-lasting properties would pay off, and it looks like they did.
They did. And they did another trial with men and gender diverse people. This was in 6 countries, the US, I think some South American and Central American countries as well. And again, the reduction that you would see with lenacapavir, how likely it is of an infection, was massive. So it was 2 people out of thousands who contracted HIV versus I think more than a dozen or dozens in the other groups. So it's, again, this time their estimate was that there was 82 to 99% efficacy of this drug. And again, they saw that Descovy and Truvada had a very little impact compared to the background rate.
It's a big win. Okay, so there were 2 cases, so it wasn't 0, 2 out of a couple thousand, but the reduction in risk is enormous.
Is huge. I would say that you shouldn't go away from this thinking this completely prevents infections. There is still a chance, but the reduction is so large that it is a really important breakthrough.
And my hope, I hate to be hopeful, but you can get non-linear population effects with transmission reductions, where if transmission per event drops 90% at a background rate, the background rate might start dropping too. I mean, depends on interaction with treatment drugs and a lot of other factors, but if you can imagine that per event your risk is going down, and then over time the background rate is going down, that's actually a very large effect together.
That reminds me of this concept of the reproductive number that a lot of people would have heard about during the COVID pandemic. So they are not. So this is the number of people on average that someone infects if they've been infected. So if I was infected, maybe I would infect 3 other people on average with the coronavirus. In this case, yeah, so the higher the number, the harder it is to control the disease, but also the faster it spreads in the population. And if it gets below 1, if I'm just spreading it to less than 1 person on average, eventually that disease will die out.
It's a dream that we can head towards now, maybe. Okay, that's PrEP. Did Lenacapavir get tested in trials for treatment as well, not just prevention?
Yes. So it was actually tested and approved as a treatment drug before these preventive trials and results. The first trial was as a treatment for drug-resistant HIV, where they're testing lenacapavir plus the standard regimen that people are having in people who have tried many different treatments so far, and this is Plan C or D.
Right.
And so it was effective in those trials. It was approved as a treatment for drug-resistant HIV based on that. They also did a phase 2 trial where they tested it as a first-line treatment.
Okay.
So that means that would be how effective is it as the first treatment that someone receives if they have been diagnosed with HIV? So they compared lenacapavir with other existing drugs. There are a bunch of other ongoing trials still. So I think there are more long-acting treatments where they're testing a combination of lenacapavir and islatravir, which you mentioned. That was the Merck oral pill, which are both long-acting, right?
Yeah.
And then there are a bunch of other types of treatments. So they're doing testing in children and adolescents. They're also testing whether it can be used in people who have been receiving other types of antiretroviral drugs, but they still have some HIV that's suppressed in their body. So I think, as we talked about hours ago, one of the things that you see with HIV is that these drugs can block the replication, the multiplication of HIV in your body, but there are also particles that would stay in some parts of your body, kind of hidden and silent. And these reservoirs of HIV are difficult to get rid of. So is it possible to use lenacapavir to disrupt these reservoirs? That is one of the questions that they're looking at with another trial. And then there are a bunch of others. So I think there's another one with lenacapavir plus cabotegravir in people who have taken lots of other treatments. And these trials are being conducted essentially almost like all over the world. So it's North America, Europe, and Southern Africa, many different countries. It's obviously quite a big process to do this lab testing, to develop these drugs, to then test them in some places, in certain countries like the US or like Southern Africa, and then to scale it up to these massive trials is quite interesting, but also to me quite impressive how fast this has happened. The drug was only developed in 2018,
Wow.
and it's been, well, okay, it has been 7 years since then. I'm used to seeing timelines that are so long. And this was approved for the first time in 2022, I think, as a treatment for drug-resistant HIV, which is
Okay.
only 4 years in trials. I think that, I mean, on a personal level, I think that could still be sped up, but like that is an impressive speed.
That's a 4 year starting clock to finish line. Not so bad. And but we're not at the finish line yet. The finish line is this really going to impact people's lives who are at risk of HIV? So just summing up what we just covered, we have now looked at long-lasting drugs as a concept and other HIV long-lasting drugs and, long-lasting drugs in other areas that we're excited about, but are still in development. And then you just outlined with lenacapavir, what was the clinical story to get here and who can benefit from lenacapavir. And it sounds like the people who've called this a miracle drug, to me, are basically right. This, in the clinical trial for cisgender women, there were 0 HIV infections among the 2,000 women who got this injection, and there were tens of infections in the other arms for women on other forms of PrEP. So this is a totally different situation. And for men who have sex with men and other trans people who are in the other trial, the other phase 3, there was also a massive drop of, say, 80, 90 more percent in transmission. So how do we get this drug to people, Saloni? That's what I want to know.
Maybe also just to think back about the whole timeline of drug development in this field. In the 1980s, in the early 1980s, when the first case was reported, no drugs. People thought this was an untreatable disease, or they wanted to treat it, but they had no idea how. 1987, the first HIV drug, azidothymidine. 95, the first protease inhibitor, and the start of combination therapy that completely changed the survival for people with HIV. In 2012, is that right? We have PrEP
Truvada.
introduced. And then in 2022, we have lenacapavir as this drug-resistant treatment. And then now we have lenacapavir as a preventive drug that is so long-lasting and both a breakthrough in terms of the effectiveness, in terms of how you take it, how long it lasts in the body, but also because it was a completely new type of treatment. It inhibits the capsid of the HIV virus. It's not just tweaking existing drugs. It's this whole new type of treatment that now opens up the field of research to developing more capsid drugs, I think, as well as more long-lasting drugs in the body.
All of that building on each other to get us to this moment. The decades of science. I feel so grateful.
Decades of science. Let's talk about where we are now in terms of, or how are we going to scale this? I mean, not us specifically, but how are people going to scale up this
You
drug?
and me, back of a van.
Getting them to everyone who, like, just driving this van around in small villages.
I'm ready. Road trip?
So, how is this going to be rolled out to people who need it across the world? I think, I mean, the most important continent here is Africa and Southern Africa. Maybe we should talk a little bit about what has the situation been like until now? How does that process work? How are people getting treatments across Africa, and how has that happened?
I'd love it.
Let's talk about HIV treatment and prevention around the world, how that's worked, so far, where we are now, where we could go from here. I didn't know until a few years ago how big the HIV treatment and prevention programs were worldwide. So the biggest program is PEPFAR, the President's Emergency Plan for AIDS Relief, which was launched in 2003 by the Bush administration. That was, at that point, the largest ever U.S. global health initiative for a single disease. And it was $15 billion as a commitment over 5 years to fight HIV and AIDS in affected countries, mostly in Southern Africa, but also other countries. I remember reading about how this was formed, how the whole program came together, and it was super interesting and inspiring. This idea that you could actually set up this huge program to treat millions of people in the poorest parts of the world with this against this really deadly, scary disease. And at that point, there were effective combination drugs available to people in the US and other richer countries, but people in Africa were not, they weren't able to access them,
Yeah.
which is quite scary. You have such a hugely unequal outcomes just based on where you live, but also there is this drug that feels just out of reach that wasn't getting to people.
Yeah.
And what I read was that Bush wanted to do something big on HIV and AIDS, and he asked several people working with him on health in the US, including Anthony Fauci and Dr. Mark Dybul, to figure out what was possible. And they looked at what was already being done in Africa. Was there anyone who was receiving treatment at this time? How were they getting it? And the main source that they found at the time was TASO, the AIDS support organization, and Doctors Without Borders, or Médecins Sans Frontières, and they had been providing treatments to people. Um, I think it was two countries, South Africa and Malawi. They were providing these generic versions of antiretrovirals to them on a voluntary basis.
Right.
But what really stuck in my mind was when I was reading this interview of Mark Dybul, one of the people who worked on setting up this program and planning it out, was that he mentioned that at that point, also the AIDS support organization were actually transporting this in little, I think, fridges on their bags, um,
Wow.
that they were carrying around on motorbikes around, to remote villages to get these drugs to people who needed them. That was very inspiring. But the success of Doctors Without Borders in these small scale, providing treatment at a small scale, showed that this was possible.
The
Could it
proof
be
of concept.
scaled?
Yeah.
It's a proof of concept. So now what's needed is to set up these supply chains to do this at a much bigger scale, to set up the drug development and manufacturing, set up the networks of clinics and the people who would be providing treatment to people in remote villages, and so on. And so Mark Dybul and Anthony Fauci put together these plans of how much this would cost, what it would look like to operate this, how it might look like at scale. Um, the other reason this is really interesting is because at the time, people didn't think it was possible to do this. They thought this was just some pipe dream. Several reasons. One is this is a really poor region. Uh, Trying to set up something like this at scale requires a lot of work. You have to work with community leaders. You need to hire people, train people to provide this treatment. I think there was also this perception that some people had, was that people, like poor people in Africa, couldn't take daily pills. They couldn't follow these regimens. And I mean, the fact that this program is so effective, I think, has shown that that's not the case. But it's also this idea that, just because something is difficult to take regularly doesn't necessarily mean that we should stop there and accept that as, status quo. You could eventually develop long-lasting drugs. You could find some way to make it easier for people to access these treatments on a regular basis. And I think that kind of attitude shift is really important here.
Yeah, you can do big things and sometimes they work. And at the turn of the century, I feel like there was more optimism around, big global health improvements and projects. And at the same time as PEPFAR was getting started, or at a similar time, the Global Fund was getting started, which was not just a US program, but was a multilateral program that involved many different donor countries, so higher income countries contributing into a pooled fund, which would focus on HIV/AIDS and malaria and tuberculosis, three of the biggest infectious disease killers around the world at the time and still now. And since then, we have, as a species, as a global society, made a huge amount of progress on all three of those diseases. And, we should probably show a graph of HIV/AIDS incidence, but also AIDS deaths, because the curve really bent down with these commitments from the Global Fund and from PEPFAR. It was a problem that seemed to be spiraling out of control. And then lots of energy, focus, attention, resources were put into it on really scaling up proofs of concepts and really committing to it. And lo and behold, there were results. And that's inspiring to look back on. And I wonder what it would have been like to be in the room when people thought it was impossible and you really thought, like Mark Dybul and others, well, we got to go for it.
It's also incredible to think about, from the perspective of people in Africa, how common HIV was at the time is probably not obvious to some of us. But in the year 2000, there were several countries in Southern Africa where some 15 to 30% of the adult population had HIV. That is scary to think about for such a deadly disease, how it affects, the people themselves, their families, the society as a whole. This was really effective and successful, both at changing what life was like for people with HIV, but just also the culture around it. And it's continued since 2003. So it's just an incredible program. It's one of the biggest global health programs, but at the same time, it costs a very small fraction of our incomes here or in the US to contribute to PEPFAR or the Global Fund, and it makes a massive impact on people around the world.
Well, it reminds me of the graph that you showed earlier in this episode when combination treatment first came out in the 90s, and you saw this totally discontinuous drop in mortality rates. And this was scaling that drop up to people who did not have access to the drugs until there was a global commitment behind them. And that means that there are many people, not just millions, but tens of millions of people who are alive right now, who are on drugs that control their infection, who would not have been alive. And it's so heady, it's impossible to, at least for me, to get my head around that. But friends, so many families. So it's mind-blowing to think how different the world would be for so many people.
The scale of this is also incredible to me. Like, the estimates are that there have been 25 million people, whose early deaths were prevented because of PEPFAR as a program. 25 million is such a huge, in terms of the number of lives saved, it's just so huge to think about. It's like London's population is, 8-9 million. That's more
Over
than
2 times
two
more.
of the entire. Um, wow, it's just imagine that not existing and all those people not being alive.
Yeah.
It's just a huge impact.
20 San Francisco's.
20 San Francisco's.
So what's happening now then? It's all good news by the sounds of things.
It's not good news, sadly.
God.
I mean, it's April when we're recording this, and there's in the last few months the picture around PEPFAR and various other global health programs has completely changed. In at the end of January, there was a foreign aid spending freeze, so all funding to various global health humanitarian programs was frozen. Also, staff who were working at the U.S. aid agency, USAID, were some thousands of them were laid off. The remaining ones were asked not to talk to the public, but also they weren't able to keep in contact with the programs in the field. They were just banned from communicating. There was also this, because of all the layoffs, that also meant running the programs on the field was difficult for people who were working overseas. And this freeze was meant to be, this is the stated intention, which is that it was for a 3 month long review of these programs, of all foreign aid programs, to see if they aligned with the Trump administration's interests, such as national security and things like that. But a lot of them were, I mean, this just froze all of them at once rather than doing a review at the same time while they were ongoing. But also, even after this freeze was ended, which was after, I think it was 39 or so, 35 business days instead of 3 months, and some 80 to 90% of programs were just canceled. Because programs refers to specific recipients of funds, I think, for specific purposes, that doesn't mean that that PEPFAR was cut by 90% or so. For HIV and AIDS, I think it was around 23%, if you roughly estimate, based on the amount of funding those specific canceled programs received, that were cut. And that seems like maybe just a fraction of this worst case scenario of the whole thing being paused. But I think because so many thousands of staff were laid off, and because of this uncertainty and this freeze, that actually had, from what I can understand, quite a large impact. One reason for that is clinics or programs receiving the funding, some of them weren't able to survive more than a few months without continued funding. Some of the funding was for stuff that they had already completed and they were just waiting to receive the payment for it, and that was canceled as well, because some of them were for continued work. This was canceled. So if clinics are not able to survive for more than a few weeks or a few months without this funding that they're expecting, they might just shut down. And so in the field, what people might have seen would be, a physical clinic that's there. Sometimes the treatment is actually inside the clinic, but they can't access it because the clinic is shut down, and or the staff is just not around. So there aren't people working the clinics. And thirdly, trying to get new supplies to these clinics was also disrupted, so they weren't able to restock on a lot of the treatments. So at the point where we are now, I think there's been a lot of disruption, but there's a rough plan to merge all of these foreign aid programs under USAID into the State Department of the US. I think this is still a little bit up in the air in terms of how that will actually work. How will it affect PEPFAR? One thing I'm slightly worried about is, what will they actually cut? What aligns with the Trump administration's interests and what doesn't? And I think you can get a little bit of a clue from how they responded when this funding aid freeze began in January. they initially said that there were certain parts of the program that would continue, such as treatments for pregnant women. But from what I understand, that actually didn't happen in practice because of all of these layoffs and the funding cuts and so on. But what I'm worried about is, what about prevention as a whole? We think about the critical part of PEPFAR as being, preventing mother to child transmission, but it's also this, the broader thing of how do we reduce
Yeah.
the spread of HIV? How do we treat everyone who has HIV? And this began as such an ambitious and effective program that managed to treat some 20.5 million people last year, and now that's massively been frozen, and we don't know how much of it's going to remain.
It's so frustrating. It's so frustrating to see such a good use of money that in the grand scheme of things is not that much money from the US government's perspective. I mean, it's frustrating in the short term. I'm more nervous about treatment than prevention of what you're describing of clinics being closed. If you have HIV, you need to be on daily drugs. And if you run out and can't get a refill, oh God, it must just be so scary. It must be so scary. And then long term, I totally agree. We're at this wonderful moment of prevention, and driving down transmission is just becoming more and more possible with lenacapavir and with other drugs. And I hope
I
that.
find it no,
Go
I
ahead.
find it really heartbreaking because until this point, until this year, I was so excited about, how would lenacapavir or other long-acting treatments change the picture? How, like, would we be able to effectively eliminate the transmission of HIV in some of these countries? And I think that's possible. And I think that's, a little bit ambitious, just like PEPFAR is. I think it's possible. But instead of going ahead and kind of going big on this, trying to really cut it down and actually then not needing such a large program because you managed to reduce the number of people who are affected by this. Instead of doing that, we're on this road of a lot of uncertainty and disruption, and that it's come so suddenly that people couldn't plan for it. They didn't
Yeah.
expect that their treatment would suddenly disappear. I think what the figures were showing that I was reading was the program PEPFAR is so big. There's 20.5 million people on treatment, receiving treatment from PEPFAR per year. On a daily level, that's some 200,000 people who are getting their refills. 200,000 people who are realizing
Wow.
the
Yeah.
clinics are shut and they don't know how they're
Oh
going
my
to
God.
get their next supply.
Ooh. Well, and that's 200,000 cases of higher risk of mutated viruses as well, presumably.
A risk of the resurgence of HIV if people stop taking treatment for I think it's a few months or so, and that has some quite nasty side effects in the initial period. And then there's also all of the complications that people would have with HIV as a disease itself.
Okay, so to summarize, the launch of PEPFAR, the launch of the Global Fund were a particularly ambitious moment, and there were people around who thought maybe they would not achieve their aims because it'd never been done before. And lo and behold, they achieved great things for tens of millions of people. Now
For over 2 decades.
over 2 decades. And now we're not only, we've been talking in this episode about great technical achievements that could be a launch of a new ambitious program and really drive down transmission, but really we're at a state where not only are the ambition levels lower, but the basics are not, as we record, being provided for everyone who needs them.
Right. It brings me to this point that I often think about. So I write this Substack newsletter on medical innovation, and what is the use of new treatments if there's no one to distribute them, or if there's no one to that can access them. This is the whole point of medical innovation, or like these breakthroughs don't matter if they're not getting to the people who need them.
Okay, so do we have any hope for the Global Fund? So listeners who are in the US, think about PEPFAR. Listeners in other countries, think about your own health systems and think about the Global Fund, which I assume is up for replenishment pretty soon and is doing a lot of this work too.
Replenishment is when they get funding replenished from various countries.
Yes, exactly.
And those are decided by their foreign aid budgets.
Correct. So foreign aid in many higher income and middle income countries will contribute some amount of tax revenues or of government revenues to foreign aid to other countries. And a good slice of that is global health, and HIV treatment and prevention is a reasonable portion of that global health contribution, often via the Global Fund. And the UK is not in a particularly ambitious moment either. At the time of recording, we are a month in, or possibly 2 months now, into an announcement under the Labour government that instead of returning to 0.7% of GNI, basically GDP, being contributed to foreign aid, as was the case under the last Labour government and the beginning of the last Conservative government, we are going to drop down to 0.3. So under COVID, we dropped from 0.7 to 0.5 in what was termed a temporary measure for COVID. And now, instead of returning to 0.7, we're dropping down to 0.3, and we're going to
30%. Wow.
Yeah.
So small. I mean, to me, it seems so small as well, because I like have read about some of these other programs that our foreign aid has funded, and some of them are really impactful. It's stuff like,
oh my gosh,
it's vaccination of millions of children against these deadly diseases that don't really affect us very much in wealthier countries. It's stuff like trachoma, which is this bacterial eye infection. The US and the UK and some other philanthropic donors came together to fund this really ambitious program to supply antibiotic treatments, better sanitation measures, and so on to hundreds of millions of people across Africa. And they've massively reduced this, really painful bacterial infection that can lead to blindness in children. These are some huge successes that many people don't even know about.
tuberculosis way down from 20 years ago. Malaria recently stalling, but has been driven down a lot since 2000. Mostly affects children. Progress is possible. Okay, well, now that we've depressed ourselves sufficiently, there's both the financing that is not looking so hot right now, the ambition not looking so hot right now. Another thing that you can do to try and, get more people access to treatment and prevention is to drive the cost down of the actual drugs themselves. And maybe it's time we talk about that with respect to Lenacapavir.
What do we know about the cost of Lenacapavir right now?
Well, there's the cost of production, and there's the price that the company selling the drug charges. The price that Gilead is charging in the US is $42,250 is the last number I saw.
Per person.
Per person per year. So that covers all the injections for the first year. That is about double what cabotegravir, the Viv injectable, is priced at in the US. I was priced out of cabotegravir. I assume I will be priced out of lenacapavir in the short term. Why do they charge that much money? Well, a lot of the development that we talked about earlier in the episode is done as a pure expense. So all of the scientists who are working at Gilead trying to iterate on the drugs to make them better, the funding for all the, or at least most of the clinical trials we discussed, that's all done as an expense. And so they want to charge more out the other end to some patients so that they can recoup some of that money. And if they make any profit, hopefully some of it will get reinvested in more drug development, not just distributed to shareholders. That, of course, raises questions. Well, do you think that someone in Botswana is going to pay $42,000? My guess is no, and I'm not going to pay $42,000, so I'm with them. So the price versus cost is very different for patented medicines often. And there are estimates for how much it will cost to produce a generic version of Lenacapavir that are under $100. I've seen as low as $40. I haven't looked into the drivers of those estimates or how much Gilead has really revealed about the production methods in public, which would give you better methods of coming up with estimates there. The good news, though, is that Gilead has already signed agreements with 6 generic suppliers of Lenacapavir for 120 countries. So plenty of lower income and middle income countries in that mix. And you may recall there are about 200 countries in the world. So there's a lot that are not covered there. But for those 120 countries, these suppliers will be able to provide versions of Lenacapavir that have been shown to be therapeutically equivalent to the initial drug that Gilead used in the clinical trials. Though these new the new production runs of these will be made by different companies that have been kind of given the rights and taught a bit by Gilead how to do it. And those generic companies will be manufacturing, hopefully, eventually, enough supply for use in those 120 countries. And what Gilead in their press release in October has committed to is that they plan to provide Gilead-supplied product at no profit to Gilead until generic manufacturers are able to fully support demand in high-incidence, resource-limited countries. So that's a great start, to be honest. The questions that leaves me are, well, how quickly, what is the supply? How quickly could demand be met? And are we sure that they're going to ramp up quickly enough? I mean, number one. But then secondly, what about the other 80 countries? So there are plenty of countries in South America, for example, that are not in the 120 covered by this generic agreement and that have relatively, have medium HIV incidence where a lot of people could be protected by this drug that now exists.
Some of them were part of the clinical trials for lenacapavir as well, which is quite depressing. But I think what I've read is that they plan to provide it to people in the trial. I'm not sure that extends to the whole country. I mean, could someone just make this drug themselves? Or, I mean, not like an individual person, but like, imagine there were some other manufacturers that weren't signed on to this agreement.
There's something beyond that, which are global intellectual property and patents and enforcing those. And then there may also be some technical barriers. So I'll talk about the patents and then the technical barriers. So on the patents front, there's a long history with HIV specifically of the tension of global pharmaceutical companies who want to enforce patents and high prices and patients and activists and advocates who want medicines to be available for more people sooner. And there's two broad solutions to this problem. One is what I just described with Gilead, which is voluntary licensing, where Gilead, arranges with other generic companies, okay, you can do this and we will not sue you for selling these drugs in these 120 countries. But if you sell them elsewhere, maybe we will sue you. And then so that's voluntary, and there's then compulsory licensing, where a country may determine that they have a public health crisis to the extent that they are not going to, the normal patent rules are out the window. So this almost happened with HIV in South Africa in the late 90s, early 2000s, and more recently, I believe, in Colombia, where the government's like, make drugs, that's okay, so long as people get the drugs. And I think that is appropriate in some medical emergencies, and it was a big topic of dispute and debate in COVID as well, where I think the debate actually goes a bit of a different direction, probably not worth getting into now, but vaccine manufacturing is quite different to generic small molecule manufacturing. The small molecules are, in general, pretty commoditized. There are many, many companies who can make them in many, many countries. And vaccines, the product is the process to some degree. How you actually manufacture a given vaccine in a particular bioreactor with particular cells, with particular growth medium, you got to kind of get taught by the original manufacturer. It's harder to just scale up, and it's harder to even infringe on a patent if you wanted to. So that brings me to the technical blockades here, where for a traditional generic drug, a small molecule, there are not many technical blockades. You can just, even if a company has not revealed all the secrets of how they made something, they have to file with a regulator and give some information, some of which is then made public once a drug approval is given. They have to list certain information on a label for patients of what the heck is in this drug. And also, if you're a competitor, you can simply buy their drug once it's on the market and analyze what's in it. So you have a lot of tools where you can basically enter as a competitor from a technical point of view. Then the difficulty with some long-acting drugs is that they are more complicated to copy. And so if you were dealing with a long-acting drug that had a liposome or had a particular polymer.
What's a liposome?
I don't want to answer that question because I'll get it wrong.
It's a fatty blob, right?
Say it again.
It's a fatty blob.
Exactly.
Lipo
Thank
just
you.
means
It's
fat.
a fatty blob. I think it's probably a bilayer. I think basically imagine a fatty blob that encapsulates the thing you care about. But there are different fatty blobs you might want to make, and there are companies trying to improve their fatty blobs. And it's harder to copy the fatty blobs that are right at the frontier of fatty blob technology than it is to copy the small molecule. Now, the good news about lenacapavir and the good news about cabotegravir and the good news about islatravir, 3 of the long-lasting drugs we've talked about for HIV, is that they don't seem to be right at the hardest end. We don't have a liposome, for example, involved. We do potentially have some things that make it a little harder than usual. And I'm a little bit beyond my knowledge about how it applies to lenacapavir, and I would love to see how it goes. But for example, I think you need a nanomiller where you grind up your drugs. So this is true for cabotegravir, and I think probably not true for lenacapavir. You grind up your drug crystals so they're tiny, 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 so that when you disperse them in a liquid, and then when you inject that liquid with the solids, you've got solids that are high surface area to volume ratio. And so the question that comes to mind for me is, which generic companies own a nanomiller, for example? And does that machine cost $8 million? How much? So those questions start rearing their head. And the next set of questions for me is, there's a tried and true regulatory pathway at the FDA and other regulators for generic equivalents to small molecules. You have to prove only that they are equivalent in certain respects. You do not have to redo everything else.
Mm-hmm.
What are we sure that those tests are going to be good enough for long-acting injectables or for long-acting drugs in general? Or is there some reason why towards the tail end of many months, there may be more deviation than you got from just testing the batch chemically? And, well, should we actually therefore rerun a big clinical trial? And that would be cost prohibitive. Then you really would not see much generic entry. I don't think that that's the way lenacapavir will go. It certainly would get my hair up if people started worrying about it, though.
I mean, I guess this also makes me think about Gilead producing it, probably with multiple manufacturing plants or so, and they would have to do this internal testing, presumably. Hopefully, there's a way to do that in the same way, or they're producing it in a very similar way that they would be able to know early on. Is this the equivalent? Are these molecules equivalent across these different sites? The other question that I had was I had heard of this thing called the medicines patent pool. Is that
Yes,
something
yeah.
have we already covered that, or what is that?
Well, conceptually, a little bit, but actually, no. So they are kind of an intermediary. They're a UN-backed nonprofit that tries to help match up originator companies that are filing patents on new medicines, taking those medicines through clinical trials, marketing them in some countries, trying to match them up with generic companies who might want to market the drug in countries the originator company doesn't focus on as much or doesn't care about as much in terms of, making a profit, for example. And often there's a match to be made there that both companies are very happy with. Especially if you're a smaller originator company who doesn't have any experience selling into Bangladesh, then you might very well want your drug to be used by people in Bangladesh who want your drug, but you just don't have the resources to get up to speed with the drug regulator in Bangladesh. You're going to be on the hook for if anyone sues you for side effects in Bangladesh. It's just it's a big proposal, but the medicine patents pool MPP will sit in the middle and say, look, we have relationships with many generic manufacturers, many of whom have lots of experience in Bangladesh. If you just sign on the dotted line here and say that, you're not going to sue them if they sell in Bangladesh, then you can both be happy and patients get to benefit. And they've had some successes, in particular with HIV over the years. I think they were set up in 2010, so for the more recent round of voluntary licensing, but they worked on cabotegravir, the initial long-lasting injectable drug, and sort of sat between Viv, the originator company, and I believe 3 generic companies there to help transition that, help get that out into more countries.
I didn't know much of that at all. That's really cool. I'm kind of thinking about, okay, we heard a little bit about how PEPFAR was formed and that was set up. I'd be very curious about that. Um, the other thing I was thinking was, okay, given that we have all of this, we have the medicines patent pool, we have these licenses with generic manufacturers. Also, Gilead I think they've said that they have the capacity to manufacture up to 10 million doses this year. I initially thought that was a big number, but if you think about it, it's 2 doses per 6 months. So
Divide by
divide
4.
10 million by 4, that's roughly 2.5 million people who would get this. And it's both in terms of a treatment for drug-resistant HIV, and it's going to be used for preventive. So that's actually a small fraction. And so hopefully the generic manufacturers help to scale that up to some degree. But aside from that, okay, if PEPFAR has this uncertain future right now, I think we do know that the Global Fund is going to try to roll it out, but trying to scale that up is, I think, maybe the next big thing to try to focus on if people are listening and have some way to convince their government that this isn't a really important thing to work on.
Yeah.
So I think Global Fund seems like it's going to be doing lots of the work in terms of funding these programs to roll it out and administering it worldwide.
Yeah.
But I'm curious if there are other things that come to mind in terms of how you're thinking about the potential future here.
Those are the main ones to me. I think the time is now and the opportunity is here. And, if you really just take a step back a whole nother level, what is happening in many countries most affected by HIV? Well, a happy piece of news is that I'm cautious saying this in April 2025, where trade relations are also a little bit up in the air. But the happy news from the last few decades is that most lower and middle income countries have been growing economically. And that was not true in the 1960s, for example. We in fact, in most countries have been seeing income growth for people and seeing the tax base of those countries also grow, meaning those countries can do more public health interventions too. And, that is really the future that will make a lot of people with HIV have more sustainable healthcare, is that if you're in Nigeria, you should not have to rely on the whims of the American public. And in the future, you hopefully will not. But that, unfortunately, is decades away. The tax base of many countries is not high enough to provide lenacapavir to people who need it. And people are not individually rich enough to pay for lenacapavir if they need it. So now is a moment for people, especially voters in richer countries, to kind of do our part. It's not a permanent humanitarian effort. I think it's a decadal. It's, the next few decades really matter, and we have these amazing new technologies that actually enable a dent to be made. So that would be where I would leave the topic of scale up. Is now's the moment, and I hope that when you and I are 30 years older, this kind of topic doesn't exist in the same way, because we don't have to think about external sources of financing quite as much.
Oh, I really hope so as well. I mean, I think right now where we are, this still seems quite out of reach for a lot of people in the most affected countries in Southern Africa. I think I was reading the average spending on healthcare is some $80 per person per year in Southern Africa, whereas it's, what is it, like 80 times that or something in the US?
Right. So, yeah.
And meanwhile, I mean, that's not just for lenacapavir, which would be some $40 or so at a generic price. It's also all the other treatments, preventions, testing, and so on. Right now, it still feels quite out of reach, and I'm just, the Global Fund is going to be really important, trying to keep PEPFAR running, but also trying to build up more capacity within these countries to protect the people who are affected by HIV right now. That seems like a difficult problem.
It sounds like there's some uncertainty in the next decade then. So are there other tools that are maybe not scientific, but more economic or financial, that we can apply to be more ambitious?
I think there are a few. I mean, it's not just lenacapavir. I think this is one really important area that we want to scale up. But there are also these, potential future drugs like islatravir, any other potential long-acting drugs that might work. How do people get the funding model right so that we're not having to be in the situation where a pharmaceutical company is trying to recoup their costs at a very high price in richer countries, and then hopefully voluntarily agreeing to these agreements, which may or may not be scaled up. One idea that comes to mind for me is this idea of an advanced market commitment, or an AMC. And I love the idea of AMCs. I've written about them a bunch. I've read a lot of stuff about them. I think the way to think about this is to contrast it with the regular approach to funding drugs or vaccines. So we usually have this situation where a pharmaceutical company or a philanthropic funder or some government is trying to decide upfront which bets might work out, which companies or which researchers might develop an effective drug, and they're funding those groups directly. And then some of them will work out, some of them won't. in drug development, the success rate is very low. And that means most of these bets are going to fail. There's going to be a lot of wasted money on the part of the funder's side. And secondly, there's this huge expense covered by individual pharmaceutical companies in developing the drug that they now want to recoup. And so they charge these very high prices. Takes a while usually for them to for a drug to go off patent or for them to agree to these generic licensing approaches, and that is a lot of time wasted. It's a lot of people who are not getting the drugs or the vaccines that they need.
Mm-hmm.
Is there another way to do it? And I think AMCs are one answer to that. And AMC is kind of an inversion of this, where instead of funding the groups directly, you're funding the potential successful products at the end. So you're setting up this pool of funding, which might be, some billions of dollars or so. If a company or a research group can develop a drug or vaccine that meets certain standards, and the amount that's given to these companies or manufacturers depends on how much they're manufacturing. So it's usually on a per dose basis, essentially. How many doses have you administered? You get more funding based on that. And I think this is a really cool idea for two reasons. One is, as a funder, you don't have to know who is going to succeed. You have this pool of funding if something succeeds. If nothing succeeds, you don't pay that money. So you're saving on that. And secondly, you're also rewarding companies that scale up the drug faster and get it out to people who need it. At the same time, you're only doing this for the successful drug. So you're giving this stable potential future market to companies. They're going to have this commitment in advance, often years in advance, of what the price is going to be, and they can plan much more effectively based on that. This has been tried for pneumococcal vaccines in the past. I think this was in the late 2000s. There was this advance market commitment set up to try to speed up the production of pneumococcal vaccines. So pneumococcal disease is a respiratory lung infection that affects people worldwide, and we already had effective vaccines for it in richer countries. But in Africa, there were different strains of the bacteria that weren't targeted for the vaccines. And so this AMC was set up knowing that it was possible to develop a vaccine for these other strains. And there was this pool of some $1.5 billion that was there for companies to receive, depending on how much they produced, if they managed to get a vaccine through clinical trials, show safety and efficacy. And it was very successful. So the scale up of this pneumococcal vaccine in African countries was very fast. I think 3 or 4 companies managed to produce effective vaccines, including the Serum Institute, and I think Pfizer was another one of them. And it just shows this model of how this can work. And you don't even need to have it for, you don't even need to believe that it's possible to develop a drug or vaccine for it, because if it doesn't work out, you don't have to pay that funding out. The people who do have to pay are other, like the pharmaceutical company themselves. In the early stages, they will still have to make the decision on whether this is a good bet for them.
Yeah. I think in that case, in the pneumococcal case, the Pfizer vaccine did, people reviewing what effect did this really have. They think that did get rolled out quicker, maybe scale up years quicker than it would have otherwise. I think the serum one ended up coming through later and maybe being less effective, but the scale up is so important for actually getting drugs to people who need them, not just inventing cool stuff. And when I think of applying this across to Lenacapavir, I think to this great piece that Kamal Nahas wrote in Asimov Press about Lenacapavir, and where he touched a bit on the voluntary agreements in 120 countries and what's happening outside of those countries. And, maybe this is the shape of problem that for those 80 countries where there's not maybe in each country enough demand, or there's too much uncertainty around demand for a company to, like Gilead or a generic company, to enter that market and try and start selling to the public healthcare systems. If there were an AMC that aggregated across those countries and made the demand clearer and had a price that was, fair, but also enough that money could be made to make it sustainable for the companies entering. Maybe that's this place for an AMC. I don't know. What do you think of that?
That's a great example of where it can be used. I actually also think it could be used in scaling up a drug even once it's been approved,
Yeah.
because the second part of what an AMC is used for in the scale up, having the amounts that companies receive be based on the amount they manufacture, means that you're incentivizing this large scale manufacturing and actually administering it to people. And that is something that could still be used even now.
Yeah.
But one of the other applications is as a way to pull funding towards some drug or vaccine or some product that hasn't yet been made. So that would be another, option. Can we develop a drug that's better than lenacapavir, or easier to take, or so on, and funded with this new model?
Yep.
And I think this reminds me as well. I don't know if we mentioned it earlier, but as far as I know, Gilead is also trying to produce some improvements on lenacapavir that would be taken once per year instead of once per 6 months.
Imagine that. Wow.
That would be very cool. I was wondering about why they were doing this. I mean, if you were a self-interested, profit-making company, why not just stick with this already amazing drug? And it sort of occurred to me when you were talking about Merck's drug, islatravir, that's this oral pill that's once per month. If someone could choose between an oral pill once per month and an injected drug once per 6 months, they might choose the pill.
I think
And
that's right.
not only the person themselves, but the clinics might,
Yep.
find it easier to distribute the drugs. It's just you don't need a healthcare worker or a nurse or someone to inject the drug if it's a pill. And that made me wonder, maybe that was the incentive. That was the reason that they decided, let's go even further to make this thing that's even harder for Merck's drug to beat. I don't know if that's the case, but that's what I would guess.
And it just goes to show how much progress we've made, Chevy, because when you were describing the first drugs around, they were not so good and there were no competitors. And now we have great options for patients and great options for people who don't even have HIV yet, who want to reduce their risk. So, I'm glad to be alive today.
It's so much. I mean, the whole timeline is just incredible to me. Like, to think about, we talked about, how in the early 1980s, how pessimistic or how scary it would have been to have HIV, not have any treatments, have this thought of as this untreatable disease. It's just this behavioral problem. There's nothing that someone can really do medically to treat it, and contrast that with where we are now. Like, I think personally that timeline could have been sped up. Just like reading about some of the details of early research in the 1980s, but also knowing about how long it takes to run a clinical trial, how long it takes to set up the trial sites or train the nurses and the healthcare workers, or to share this information between different research groups and so on. But at the same time, it is just an incredible story.
Now is probably the time to step back on this story that we have told and conclude. I'm sure we missed out many subplots that are also ripe for discussion. But among what we have discussed, I'm interested to hear. What were your main takeaways from this story over the last 50 years?
My takeaway, I mean, I had so many takeaways. One of them was just how many different ways you can approach medical innovation. Like, what are the different things that you could think are important here? I mean, partly it's let's make a really effective drug, but it's also how do we make a drug that's easier for people to take on a regular basis? It's maybe refining drugs that already exist, trying to improve on them in terms of their safety, efficacy, or, again, how people take them. The other was the different types of drug development. So we talked about this trial and error process with the first drug, azidothymidine, where they just, looked at 180 compounds, tried each of them in the lab, saw what worked in cells in the lab, and then scaled up based on that. The other is this is an example of kind of screening existing compounds or just compounds in nature that helps repurpose this previous cancer therapeutic that didn't work for HIV. Then we had some examples where this understanding of the specifics of HIV or how the enzymes work, what they look like, what will fit into these little gaps between them. That was another option for developing a new drug. But at the same time, there was so much iteration and
Yeah.
adjustment that was tinkering that was important there. The move from this drug that was potentially promising to one that actually met several criteria that you would have with the efficacy, the safety,
Yeah.
how long lasting it was. And then I guess there are these other, improving on the drugs that already exist that is not just trying to use existing information. So if there has been already a protease inhibitor invented, can you now develop a new one based on that knowledge? Can you develop a different type of nucleoside inhibitor like AZT? Will people develop more capsid inhibitors based on the knowledge that they have from this? I mean, all of this I think is super interesting.
I totally agree on how much the tinkering and iteration stands out as important, basically at every level. There's so much of that is happening at the screening stage. So much of that is happening, as you just said, at the designing a capsid inhibitor that makes sense for patients in a particular context. Another thing that stood out to me is how hard science is to predict, in the sense of, thank goodness, people in the last 40 and 50 years, scientists, did not only work on vaccines. We don't have an HIV vaccine, and we do have a HIV preventive drug that you can get injected with and kind of feels like a vaccine. And I'm so grateful that people were exploring different parts of the technology tree there. We don't have a cure either. We don't have a cure for HIV and we don't have a vaccine, but guess what? We have game-changing tools. And that came from a part of exploration you may not have been able to predict back in 1981.
Also, the fact that the treatments could be used as a prevention, as preventive drugs, was probably not that obvious to scientists at the time. That itself was quite unpredictable, I think.
Absolutely.
The other thing that reminded me of was just how many different aspects, or like how many different types of science, or what is involved in developing a drug is not just one person tinkering with it in the lab. It's this whole network of clinical trials that are
Yeah.
running. There's the basic research. There's, stuff like developing microscopes with a high enough resolution that you
Yes.
can really see what is happening inside the cell, what this virus looks like, what the proteins look like. There's the DNA sequencing technologies. There's the protein development, like all of this stuff kind of comes together to develop these drugs. And then there's medicinal chemistry and the pharmacology, which I think probably I had kind of underrated before, as just, okay, this seems like this last minute thing. After you've developed a drug, you now want to make sure that it's safe and effective. I previously had this assumption that that was what medicinal chemistry was about. And now I'm thinking this can make a huge difference on whether a drug is useful or effective at all in the real world.
Even right at the discovery stage of lenacapavir. So important how stable it is and how it doesn't break down very quickly. I totally agree with what you just said about interlocking parts of the medical innovation system. It's amazing that we just about had recombinant DNA, maybe at the time when HIV started becoming a crisis. So we could use recombinant DNA and in the lab as a research tool. And that we did not have all of those other things we just mentioned. We didn't have PCR. We didn't have cryo-electron microscopes, certainly. And we only in the last few years have learned the capsid, which we are now inhibiting with lenacapavir, in fact stays intact into the nucleus. There's so much more that we're still going to learn over the next coming years that might open up new frontiers. And that's true not just for HIV. I bet you that things that HIV researchers have learned will be useful for hepatitis B, for other cancers, for this, that, and the other. And, the final interlocking surprise of science that I learned from you many hours ago was that it was only 2 years before 1981, that the first human retrovirus was discovered. Thank goodness for that. And it makes you wonder what we don't yet understand that will make solving and curing diseases in the future easier.
Yes. And above all of that, the other interesting thing for me was learning about how drug pricing works, or how these patents work. How does manufacturing actually work at large scale? What do the funding models of what they have to do with whether drugs are developed, how fast they're rolled out, who's ready to pay for certain drugs. And it's like every aspect of this whole process, not just the lab, not just the clinical trials, but the funders, the people who decide that, who show support for foreign aid spending, for example. Everything comes together when we're talking about any disease, but particularly for HIV. It's so salient because of these huge programs that have transformed the lives of millions of people.
And going into the future, there's no point for all of this wonderful science unless we remain ambitious and unless we make sure that people who need these drugs can access them. It's possible. We've done it before. And into the future, we go with uncertainty and with resolve. This episode was only possible from a lot of work done by a lot of people publishing in the open, whose papers we read and whose reviews we read. I won't thank them all here, but we'll leave in the show notes some of the research that we based this episode on. I, in particular, would like to thank Anne de Bruyn-Kops, who wrote a great review of long-lasting injectables for many different diseases for Open Philanthropy that I learned a lot from. I want to thank Sinela Rankovic, who was the HIV researcher who knows all about PF74. And then, of course, I'm sure we both want to thank Douglas Chukwu, who joined us for our first ever Phone a Friend session.
Yes, and the team at Works in Progress, Arya Babu, who helps us really actually get this podcast to run, Adrian Bradley, who's here with us now, producing and keeping us on track with this episode, and then the team at Works in Progress and Open Philanthropy, who are sponsoring this podcast. And then I would say also all of the scientists who were involved in developing all of these drugs, all the people who were participating in all the clinical trials, all the healthcare workers who worked in them, everyone involved in this massive program, PEPFAR, everything. It's just, yeah, I mean,
It's so cool.
It's very inspiring.
And with that, I will ask you as listeners, if you enjoyed this episode, feel free to subscribe. We will be talking about other hard drugs in the future. And check out the show notes for more details on this one.
Bye.
Bye.